So, 6.1, photosynthetic organisms. Producers taking carbon and nitrogen compounds from the environment. Now, usually carbon comes in the form of carbon dioxide, and it comes through the stomata, which are tiny gaps, tiny holes at the bottom of the leaf. So if what we're showing right now, so if this was the underside of the leaf, you see these tiny little holes. If you use the microscope, they are stomata, and the carbon dioxide diffuses in through the stomata. Nitrogen is taken through the roots. So that's your root hair cell. Active transport takes in the nitrates. So that comes in the form of NO3 minus. Both are very important. We need carbon dioxide for photosynthesis, and the nitrogen that's in the nitrate there, NO3, that's in the soil, that attaches to glucose molecules to make amino acids. Now, alongside oxygen, hydrogen, and other elements to make small molecules. So we've got carbon, nitrogen, oxygen, which we need for respiration, hydrogen, and other elements. Now, hydrogen and oxygen together are water. So one that's missing there that should have been there is water. Water is taken in through the roots, but through the process of osmosis. So for carbon dioxide, we've got diffusion. For nitrates, we've got active transport. And for water, it enters through another type of diffusion, which is called osmosis. And small molecules like sugars, fatty acids, glycerol, amino acids, these are actually small molecules that producers make themselves. So small molecules are used to build larger molecules. So when we talk about small molecules, we refer to them as monomers. That's more, I'm explaining this maybe more from well, not maybe, but I'm explaining this from a triple perspective. Small molecules are monomers. The large molecules that they make are called polymers. For example, glucose is built up to make carbohydrates. So glucose is the monomer, carbohydrates the polymer. Amino acids is a monomer, it makes proteins, which is a polymer. Fatty acids and glycerol are monomers, they join in repetition. So we're talking lots and lots of them to join to make lipids. So lots and lots of glucose molecules, monomers, once they're repeated and attached to lots and lots of them attached to each other make carbohydrates. Amino acids, lots and lots of amino acids attached to each other make proteins, and lots of fatty acids and glycerols attached to each other make lipids. So this equation you need to memorize the word and the formula. The word equation usually comes up in foundation, and the main equation usually comes up. The main equation, as in the formula equation, comes up in the higher paper, but they can come in both. So what happens here is, as we said, carbon dioxide diffuses in, water osmosis in, and the same way when we bake bread in an oven, we need to turn the oven on. So in bread, or even a cake, let's use a cake as an example. There's my dish or my tin that I'm going to put in the oven. It's got eggs, water, milk, etc. Everything inside it. I mixed it up. It's ready to go in. Now, if I just leave it on the table, nothing's going to happen. One thing's missing. That's energy. So we put it in the oven. We open the oven up. I know that doesn't look like an oven, but just assume it is. We open it up, put it inside. We turn it on. And then the oven provides the energy for it to go from being batter into becoming a cake. Here, again, we've got our raw ingredients. We've got water, we've got carbon dioxide. The energy for it comes from the sun. So the leaves contain chloroplasts, and the green part of the chloroplast is called chlorophyll. So that energy is absorbed by the chlorophyll, which helps turn the water and the carbon dioxide into glucose. Here it says sugar, but the scientific word here would be glucose. And it also releases oxygen. oxygen which then diffuses out. Glucose dissolves into water and translocation then occurs and it takes the different parts of the cell. So what that means is we've got carbon dioxide, specifically 6 molecules of it, 6 molecules of water, and then we've got the sunlight's energy to attach them to each other to make C6H12O6 plus 6 oxygen molecules. So what I've written there is 6O2, just in case you can't see what I've written. C6H12O6 is better known as glucose. So needs to be memorized. You need to know this. Okay, so what happens during photosynthesis? So photosynthesis, as we just mentioned, the leaf is green for a reason. It's green because it contains chlorophyll. Now, if it contains chlorophyll, the chlorophyll absorbs, takes in the energy. Now, think back to chemistry, endothermic and exothermic. What does that make photosynthesis? If energy is going in, if the energy is going inwards, if it's absorbing the energy, then what kind of reaction is photosynthesis? It is endothermic. There. Endothermic reaction. So key things to learn from this slide here. A leaf contains chloroplasts. Inside the chloroplast, we've got green chlorophyll. Chlorophyll captures the light energy from the sun, and that energy is then used for photosynthesis. It's used to combine carbon dioxide and water to make glucose and oxygen. And because it's taking energy in, it's an endothermic reaction. Okay, limiting factors. Very, very, very important. And you need to understand this from a descriptive perspective. And you also need to understand this from an explanation perspective. Just exactly the same as, but similar to what we went through in paper 1. Now, those who, well, actually paper 1's finished now, but in the paper 1 live sessions that we had on YouTube, this is similar to what we went through in enzymes. So if we look at section 1, section 2, and section 3, let's look at one section at a time. As temperature increases, the rate of photosynthesis also increases until it peaks at 35 to 40 degrees. That is the description. I'm going to come back to explanations in a second. Photosynthesis rate decreases beyond the optimum temperature due to due to enzyme denaturation. That is explanation. So look, if we were describing this, initially we would say, as the temperature increases, the rate of photosynthesis also increases until we get to around 35, 40 degrees where we hit a peak. So in other words, optimum temperature. Beyond the optimum temperature, as the temperature continues to increase, the rate of photosynthesis decreases. And that's where our description ends. Now, if it says explain, we write down our description and then we say the reason for this is because. Or as temperature increases, the rate of photosynthesis also increases until it peaks at 35, 40 degrees, the optimum temperature, because. If we say because, we've explained it with, of course, some explanation. Because the increase in temperature causes reactant particles to move more rapidly due to an increase in kinetic energy. We've increased the temperature. If we increase the temperature, we've increased the kinetic energy. Kinetic energy equals half mv squared. If kinetic energy increases, the mass isn't going to increase, but the velocity is, the speed of things is. So that means the reactant particles move faster. Now again, description, photosynthesis rate decreases beyond the optimum temperature because the enzymes denature. At approximately 45 degrees, this temperature here, photosynthesis ceases entirely. That's description as well. Ceases meaning it stops completely. Why does that happen? because the enzymes have become denatured. So it's very important to have this understanding of the difference between the two. Okay, so just to clarify one thing, it's a very good, just also a very answer to a very good question. That's the optimum temperature. Now, photosynthesis is a chemical process, that requires enzymes. So at 35, 40 degrees, enzymes work as optimum level. Beyond that point, they denature. Okay, limiting factors. Let's now look at another limiting factor. So light intensity. Again, we've got step 1 and step 2. So as the light level is raised, the rate of photosynthesis also increases. So that is your description. It increases up to a point, and then after that, the graph starts to plateau. Plateau means this straight line here. Okay, and that's it. That's the description. Now, if it says explain, you write down the description, but alongside that description, you give your explanation. So as the light level is raised, the rate of photosynthesis also increases. This is because, but that's up to a point, because beyond that point, the graph starts to plateau. This is because photosynthesis reaches a limit when temperature or carbon dioxide levels become the limiting factor. To increase the rate of photosynthesis, carbon dioxide or temperature must be increased. So what's happening initially here is light is a limiting factor. So before, as we explained, we've got carbon dioxide, we've got water, we've got energy coming from the sun. Now, it might be a really dull day or it's nighttime. So at nighttime, there's no light intensity, so the rate of photosynthesis is very low. As the sun starts to rise, the light intensity increases, so the rate of photosynthesis increases until we get to a certain point, which is here, where I've put a dot here. This point here. Light intensity continues to increase, but photosynthesis is not increasing. And that's because we either don't have enough carbon dioxide, or it might be a cold day and the temperature is too low. Now, initially, because it was nighttime, light intensity was the limiting factor until we reached that point where I put all the arrows. When we reached that point, light stopped being a limiting factor, and instead, carbon dioxide became a limiting factor. Temperature might have become the limiting factor. Now, this graph looks exactly the same as the previous one, and it is exactly the same, except here we're looking at light intensity, and here we're looking at carbon dioxide levels. So if carbon dioxide levels are very low, we can't, there's no photosynthesis because there's no carbon dioxide to react with the water. But as the carbon dioxide level increases, the rate of photosynthesis increases until it plateaus again. At this point, the carbon dioxide levels are increasing further, but now something else is a limiting factor. And this time the limiting factor might be the light intensity, or it might be the temperature. So let's read through the text. As the levels of carbon dioxide increases, the rates of photosynthesis also increases up to the point the graph starts to plateau. So that is our description. And the second line is our explanation. So explanation needs both. Why does this happen? Because photosynthesis rates plateau when temperature or light intensity becomes limiting. Increasing the light intensity or temperature will increase the rate of photosynthesis. Okay, now what does this graph show? So let's look at this. Initially, what it's showing is as the carbon dioxide levels increase, the rate of photosynthesis increases up until we get to a certain point at which carbon dioxide is no longer a limiting factor. Now, if you look at line A and look at line B, beyond that point, what does this graph show us is the limiting factor? Because at 15 degrees, that was its maximum level of rate of photosynthesis. At 20 degrees, the maximum rate of photosynthesis is a little bit higher. So it shows us temperature became the limiting factor. So in the increase in temperature has increased the rate of reaction. So as the levels of carbon dioxide increases, the rate of photosynthesis also increases up to a point. We know this from the previous slides. Photosynthesis rates plateau when temperature or light intensity becomes limiting. So to boost rates, you increase light intensity or the temperature. In this case, what's happened when we increase the temperature? The rate of reaction has also boosted and increased. Okay, now with the core practical, I'm going to briefly explain this to you. But what I've actually done is I've put the link for a really good YouTube clip. Um, Now on AMALearning, we will be releasing something, but I'm not sure if it's going to be made by the time. Actually, it should be made. But even though we're releasing something on AMALearning, I still advise this video because it's made by Edexcel, Pearson's. It's got actual scientists going through the whole process. Because this is one of the core practicals, so please watch this video. But essentially, the way this experiment works is not like this picture suggests. Um, Instead of having this beaker, instead what we have is tubes. These small kind of tubes with a lid on top. And inside that tube, it's got water. It's got algal balls. So a ball made of algae. Because algae can photosynthesize, which is why they're green. And the rest of it's water. And in there's also sodium hydrogen carbonate, as in there's an indicator. And that indicator changes color based on how much carbon dioxide is released. How much carbon dioxide is taken out of the water. You see, when you've got carbon dioxide in the water, you have an acid called carbonic acid. So the water that's inside there is acidic. Why is it acidic? Because it contains carbon dioxide. Now, when the plant photosynthesizes, the more it photosynthesizes, the more carbon dioxide it takes out of the tube. Now you have a tube, you put the tube every 10 centimeters moving away from the light. Now, as it moves away from the light, what do we expect to see? We expect to see the indicator showing us something that is more and more acidic. The one closest to it, so this one here, is going to be the least acidic. As you get least acidic because it's taking carbon dioxide for photosynthesis, we expect this first one to have the fastest rate of photosynthesis. If it has the fastest rate of photosynthesis, that means it's going to take the carbon dioxide out of the water. It's going to be showing less acidity. The one that's further away, that's got less light intensity coming to it, which means it's going to be more acidic. And the indicator that's inside it will show that through the color. But like I said, please click onto this link, and that link will take you to a really, really good website. When I say website, it's sorry, a YouTube channel. The YouTube channel is run by Edexcel person, as in the company that's writing your exam. So all of the practicals that they made in a way that you will benefit from it and do better in your exam. So things to keep in mind when you're writing about this exam is your control variables. Now, the type of algal balls needs to be kept the same. So we keep the species of algae the same. The number of algal balls, because you know that little tube I just showed you with a lid that screws on at the top, you would put 10, 15, 20 algal balls in each one. So you have to decide on a number. So when you're writing the process here, you'd say, I added 10 algal balls to each tube, as in each one of these. So each one of these had 10 algal balls. They were then filled with water and an indicator. The color of the light, the best thing to use is LED. And the reason we use LED is because it doesn't give out as much heat. Because if it gives out lots and lots of heat, what's going to happen is the beakers that these tubes which are closest to the bulb, they're going to heat up. And then that's going to change the rate of photosynthesis. The temperature of the water. LED bulbs are used because they don't get that hot. When they used to use the old type of bulb, if this is the bulb, they used to put a beaker full of water in between that and the experiment. Because then the beaker of water would let the light go through, but it would absorb the heat. Now, when you put the algal balls into the water, you have to give them 5-10 minutes just to settle into the water because it's a living thing. It's been taken out of one environment and put into another one. So it still needs to adjust to that new environment. Carbon dioxide concentration. So you add the same amount mass of sodium hydrogen carbonate. So I mentioned sodium hydrogen carbonate and I mentioned an indicator. These are two separate things. I may have mentioned them as one thing. Sodium hydrogen carbonate is added to the water. So it's added to every single one of these because it releases carbon dioxide. So if we, for example, add 5 grams of it, then it will release a certain amount of carbon dioxide into all of them. So because in this experiment, what we're testing is as it gets further from the light, what happens to the rate of photosynthesis? In other words, the effect of light intensity on the rate of photosynthesis. So we don't want carbon dioxide being a limiting factor. The volume of indicator solution. So you would add the same amount of volume, same volume of indicator to each bottle. Okay, this is something that I've only ever seen in a higher paper. It's not triple per se, but I've only ever seen it in a higher paper. I'm just going to very quickly confirm this. Yeah, 6.6 is purely a higher topic. So all we're saying here is the rate of photosynthesis is directly proportional to light intensity, and it's inversely proportional to the distance from a light source. Now, we'll go through this in more detail in the pre-exam workshop. So if you're not able to attend the pre-exam workshop, please still go through all the maths questions because they are always maths questions linked to this topic. Okay, root hair cells. So what are examples of osmosis in plants? So 6.7, the actual specification point says, explain how the structure of root hair cells are adapted to absorb water and mineral ions. So this slide here fulfills 6.7. How are root hair cells adapted for the absorption of mineral ions? So it's a 6-mark question. Number one, they have an elongated shape. So what we're doing here is we're describing it, then we're explaining the benefit of it. Elongated shape, which increases the surface area for exchange. It means the actual root hair cell has all of this space to uptake in water through osmosis. It has lots of mitochondria for respiration to release energy, and that's for things like nitrates because they are actively transported into the cell, like I said at the beginning of this session. Then it's got thin membranes. So these membranes are usually only one or a few cells thick, and that provides a short diffusion pathway. That means the gas does not have to go far, or the water does not have to go far before it enters the root. So I would advise you memorize these points, 6 points, as it comes up quite often. It is, and at the end of the day, 6.7 root hair cells is a topic, is a specification point within itself. So I would advise you go through it in as much detail as you can. Right now, just to explain, so it's important we memorize this slide. Okay, xylem and phloem. What is the function of xylem? So 6.8 specifically, it says explain how structure of xylem and phloem are adapted to their function in the plant. So you need to know about the fact that xylem is lignified, so it's got lignin, and how that helps it transport water, and the fact that xylem is dead, and also that phloem is living cells, and it's got lots of mitochondria because it uses energy to transport sucrose, sugar around the plant. So here we've got the xylem cell. Here's all the key facts. It is a very common question where you get asked to explain the difference between xylem and phloem. Or separately, they might give you a question on the xylem. Separately, they might give you a question on the phloem. But these are common questions. Or you get questions linked to transpiration, translocation. When you're going through past papers, I'm hoping you'll start to see how often this comes up. Xylem, phloem, transpiration, translocation, all the time. So, number one, xylem forms a hollow tube to facilitate the easy movement of water. So there's your hollow tube there. It's a little bit like a straw. Then it's strengthened with spirals of lignin. So this lignin helps it stay strong, and that provides support and withstands water pressure. Because, for example, let's say this is a straw. Actually, I'll explain that in a second. It has no cytoplasm or organelles, and that's to ensure an uninterrupted flow of water. Because if you had these organelles, as the water is going upwards, it would start osmosing into some of these cells. So that's why it's important that the cells are dead, because the water just moves straight up. Now, if we use the example of a straw, and it's inside water. I know straws aren't that big. I know glasses aren't that small. I know water's not red. So that's full of water. Actually, let's do water realistically. So water's red. Now what happens is that straw is initially full of air. So when we use that straw, we suck all the air out initially. See if I can carefully, I'm gonna end up erasing everything. Oops. Right, so in our straw, we suck all the air out, then the water starts moving upwards. But when you suck all the air out, you have really low pressure. High pressure on the outside, low pressure on the inside. And that starts to push it inwards. And that's what happens inside the xylem. You've got a lot of pressure on the outside, trying to crush. It's actually a little bit like the paper straw. If anyone's ever used a paper straw, why are straws horrible? I mean, other than that, they lose their structural integrity when you're using them. After a certain amount of time, you leave it in the milkshake or in a drink for long enough, it's just a waste of time trying to get water up it. But what actually starts to happen is it starts to weaken as the water goes through. So the pressure causes it to crush in the middle or at the bottom or at the top. Anywhere it happens, it's horrible. So, and you wish you had a plastic one again. But obviously we don't want to ruin the earth. So it does make sense why we're using cardboard ones. But now with the plastic one is tough. It's a little bit like this lignin. So it stops it from crushing inwards because of the water pressure. And finally, the function of the xylem is to transport minerals from the roots. So the water and minerals from the roots to the rest of the plant. In other words, from the roots down here to the leaves at the top. So that's transpiration. Transpiration happens through the xylem and water only goes upwards. Okay, what is the function of the phloem? So it has reduced internal structure to create more space for transporting sugars. It contains sieve plates that allow water-carrying dissolved sugars to move freely through the plant, and it's supported by companion cells which provide energy and essential nutrients to keep them functioning. So what happens is, in the leaves, due to photosynthesis, glucose is made. And I'll show glucose as red. Now the rest of the plant needs glucose. Can anyone tell me which process? Which process do we need glucose for? Yep, well done. Respiration. We need it for respiration. So the whole plant, the whole plant needs glucose. So we use something called translocation, which only happens in the phloem, that takes water in every direction. Up, down, left, right, wherever it's needed. Now, part of the reason that occurs is because because these sieve plates. So you've got these sieve plates, and you've got companion cells that are full of mitochondria. Now, the mitochondria provides energy for, in fact, let me do it this way. Let me do the sieve plate here. So there's your sieve plate. You've got mitochondria, and mitochondria, what they do is they transport, using active transport, they transport the sugar that's on this side, they transport it to the other side. So now you've got more sugar over at the bottom half. Now, if you've got more sugar there, you're going to have less water molecules. And you're going to have more water on this side because you've got less sugar. So osmotically speaking, what's going to happen? The water is going to go from high concentration to a lower concentration through a partially permeable membrane. So that means the water will move from here to here. But why did that happen? Because the companion cells provided energy and moved sugar to the other side. So that, look what it says here, sieve plates that allow water carrying dissolved sugars to move freely through the plant. So it helps direct the water that's full of sugar. So the companion cells are very, very important. Okay, transpiration. It was 6.9 as a specification point. We need to be able to explain how water and mineral ions are transported through the plant by transpiration, including the structure and functions of the stomata. So we need to know about the stomata too. Transpiration is the process by which water moves through a plant. So it's the process by which water moves through a plant. It involves the loss of water through the stomata through evaporation. Transpiration takes place in the xylem, and the movement of water is from the roots through the stem to the leaves. So what we're saying is we've got our roots over here. Water enters due to osmosis. Now that means there's going to be more water in the roots than there is in the stem here. So water will move from the roots to the stem through osmosis. Also, what you need to remember is that this is occurring in the xylem. So now water's come from the roots and it's gone into the xylem in the stem. And that way the water continues moving to the top. When it enters the leaves, the water, The leaves use the water for photosynthesis. Some of the water is not used and it evaporates out of the plant through the stomata. As I said before, if you look at the bottom of the plant, you see what you'll see these tiny little gaps that have got guard cells around it. When the guard cell closes, it looks like this. As in when the guard cell loses water, it looks like this, so you can't see the stomata, so water can't be lost. So on a hot day, that makes sense. On a normal day, the stomata filled with water. Actually, on a day where there's lots of light, high in light intensity, the stomata fills with water. Its natural shape means the stomata then can be seen, and carbon dioxide can diffuse in, water can diffuse out, and photosynthesis can happen as normal. So here we've got the stomata. Stomata are tiny holes found in the underside of the leaves. These show individual stomata. You see this little bean-shaped cell? That's the guard cell. So when it's the intensity of light is quite high, it fills with water, and it looks like that. That's its turgid state. It's full of water, and the stomata opens. On days when the intensity is low, water exits out of the guard cells, and their shape looks more like that. So stomata allow water vapor and oxygen out of the leaf and carbon dioxide into the leaf. They control water loss and gas exchange by opening and closing. The size of the stomatal opening is used by the plant to control the rate of transpiration, because the more water that's lost through the stomata in the leaves, the more of a transpiration stream you get. Because if this leaf is empty, then the water from the stem is going to enter the leaf. Then the stem becomes emptier, and the part of the stem below it, the water moves up until you get to the roots. At a certain point, there's more water in the roots, and that goes to the emptier space within the xylem, the stem. So plants growing in drier conditions tend to have a smaller number of stomata and only on the lower leaf surface to save water loss. So if you go to the desert and you go to, for example, you have a cacti, its actual spikes is actually a rolled up leaf, but it's rolled up so tight so that all the stomata on the inside. So when water tries to escape through the stomata, it reduces that water loss. So plants growing in drier conditions tend to have small number of tiny stomata. So other plants just have less stomata and only on the lower surface. Because if something's going to osmose, let's say that's the side view of a leaf, then if your stomata on the top, things diffuse upwards. But if it's at the bottom, it's less likely the water will diffuse out and then kind of and make its way up again like that. So it makes sense for it to be on the bottom, and because it saves water loss. Most plants regulate the size of stomata with guard cells. So each stoma, so stoma is a singular of stomata. Stomata is plural. It's surrounded by a pair of guard cells. So it's what I explained before, but I just wanted to say it now. When the light intensity increases, the guard cells take in water by osmosis. So on a very sunny day, where the sun is quite intense, guard cells take in water, so water enters by osmosis and they become plump and turgid. When they're plump and turgid, they actually look more like a bean shape. So they open up and the stomata can be seen. Stomata just means that hole in the middle that I've highlighted blue there. When the light intensity decreases, the guard cells lose water. and they become flaccid, meaning they don't have much water in them. So their shape changes. And in card cells, their shape changes in such a way that it's almost like a door's been closed because it causes the stomata to close. Okay, we've done transpiration. Now we look at translocation. So translocation is the movement of sucrose. Now we say sucrose, if you get one glucose molecule, attach it to another glucose molecule, that is a simple version of sucrose. So two glucose molecules attached to each other makes sucrose. So sucrose is, in sense, as a dimer, as in it's two monomers attached to each other. So translocation moves around the sugars through the body. The dissolved sugars made during photosynthesis are transported around the plant. Why? For respiration. The sugar can be used by cells for respiration, or they can be used in other ways, such as stored as starch. It can be used to make protein. It's sugar. Glucose is also used to make starch that you have inside potatoes, etc. It is used to make cell wall. So cellulose is made from glucose. Now the sucrose sugars are moved through the phloem from photosynthetic to non-photosynthetic tissue. What this means is the glucose is in the leaves. Why is it in the leaves? Because that's where photosynthesis happens. So that's going to be full of glucose. So the whole point of translocation is take it from these leaves and take it to other parts of the plant. Other parts of the plant that need it. So that's why we're saying it goes from photosynthetic to non-photosynthetic. Sucrose is loaded into the phloem by active transport. So this is what I was explained to you before. Active transport loads it here. Now that means we've got more water here. So water osmosis from high to low concentration. So the water osmosis from a high water concentration to low water concentration through a, well, in this case, sieve plate or partially permeable membrane. And then water moves into a concentrated solution by osmosis, and this pushes substances along the tube. So that will move to there, that will push water in the other direction, and so on. Now, this is what I was saying before. Xylem versus phloem, transpiration versus translocation. These are common questions, common topics. So what is transported? So this is a really good slide to memorize. What's transported? Water and mineral ions. What's transported in translocation? Dissolved sugars. In transpiration, the transport happens in the xylem in one direction. In translocation, it happens in the phloem in multiple directions. It can go to any stem, any leaf, anywhere. In transpiration, the water goes from the roots to the stem to the leaf. In other words, upwards. It goes up. And translocation, it goes from photosynthetic to non-photosynthetic. So it goes from the leaves, these leaves don't look very good, to anywhere else in the plant. Transpiration is a passive process. It does not require mitochondria. It does not require additional energy. So additional mitochondria or additional energy. Translocation is an active process. Like we explained, you need mitochondria to provide energy for the sugar, for the glucose. Okay, this is a triple topic only. 6.11 is the only triple section of topic 6. So those that are doing combined, this should only take a few minutes, and then we'll hopefully finish this all off just before 4 o'clock. Now, adaptations of the leaf. You've got all these different adaptations. You've got cuticle, epidermal tissue, palisade mesophyll, spongy mesophyll, xylem phloem, which we've already done, guard cells, and stomata, which we've already covered as well. Now, the palisade layer, let's start with the cuticle. We'll go through this in detail again, but the cuticle is a waxy layer that stops water from evaporating through. So that means water can only leave through the stoma. Stoma means one, stomata being plural. The palisade mesophyll layer, a palisade cell is a cell full of chloroplasts. A palisade cell is a cell, a plant cell that's full of chloroplasts. So in the palisade layer, lots of photosynthesis happens. The spongy mesophyll is actually referring to where you've got less cells and more of these gaps. And those gaps are necessary that when carbon dioxide diffuses in, there's always more carbon dioxide outside than there is on the inside. And the spongy mesophyll layer provides that. Okay, so epidermal tissue, that's the outer layer of the plant, and it protects against water loss and pathogens. Palisade mesophyll contains chloroplasts and is the main site of photosynthesis. So palisade layer contains lots and lots and lots of chloroplasts. Spongy mesophyll has air spaces to facilitate gas exchange. It makes sure that inside the cell is always a gradient for the carbon dioxide diffuse in. Xylem we've covered, transports water and minerals. Phloem we've covered, transports sugar that's made during photosynthesis to different parts of the plant. Meristem tissue. Meristem tissue are stem cells, which we've covered before, found in growing regions like the tips of the roots and the shoots. And the meristem tissue is responsible for cell division and plant growth. So the palisade cells contain lots of chloroplasts to maximize photosynthesis. The spongy mesophyll cells have lots of space for oxygen and carbon dioxide to be able to diffuse through easily. Okay, back to combined and triple. So this is for everyone, combined, triple, foundation, higher, doesn't make a difference. Now the specification point here is explain the effect of environmental factors on the rate of water uptake by plants, so the rate of transpiration. So explain the effect of environmental factors on the rate of water uptake by a plant and include light, intensity, air movement, and temperature. So the rate of photosynthesis is affected by any factor which can affect the rate of evaporation of water through the stomata. So water, this is how transpiration actually occurs. Water evaporates from the leaves. So there's the water evaporated off. Water then drawn up the xylem to replace the water. So water evaporates from the leaves. Water is drawn up the xylem to replace the water. So that means water from the stem enters the leaves to replace the lost water. And the water that's before it enters that empty space. The water before it empties that empty space. And the water before it empties that empty space. And then we have osmosis, water osmotically entering the roots as well. So you have a continuous transpiration stream. So water is drawn up the xylem to replace the water that has been lost, forming a continuous column of water being pulled up the stem. As the water moves up the stem, it's replaced by water that is absorbed through the roots. So the water moves upwards, but we've got more water ready. So as the water moves up the stem, it is replaced by water that is absorbed through the roots. This works because the water molecules are strongly attracted to each other. So it explains here. Um, sorry, let me just see the time. Okay, so the different things that affect the rates of transpiration are higher temperature, increased wind intensity, high humidity, and greater light intensity. So more light increases the rate of transpiration as it causes the stomata to open, allowing more water to escape. So what can affect the rates of transpiration? So the rate of transpiration is affected by any factor which can affect the rate of evaporation of water through the stomata. So how does transpiration actually occur? Water evaporates from the leaves. Water is drawn up the xylem to replace the water that's been lost, forming a continuous column of water being pulled up the stem. As the water moves up the stem, it's replaced by water that is absorbed through the roots. This works because the water molecules are strongly attracted to each other. So what we're saying here is that water is evaporated from here, from the leaves. That leaves an empty space behind it. So water enters the empty space, which leaves another empty space. So water enters that, and then the water from the roots enters the stem, water from the soil enters the roots. So you end up with a stream, a continuous column of water. And water, one water molecule has a weak intermolecular bond with the next water molecule. So as one water molecule moves upwards, it pulls the one below upwards too. So there's a cohesion there, there's an attachment between each water molecule. Now, when we say rates of transpiration, anything that speeds up that process of the water going from the roots and out through the leaves is what we mean. This is what we're talking about. How can we affect the rates of transpiration? So number one, higher temperature. As the temperature increases, the rate of transpiration also increases because water evaporates more quickly. So what we're saying here is if the water leaves, erase this really quickly so it can be seen clearly. If the water leaves through the stomata more quickly, then that means it's going to be replaced by water before it more quickly. So transpiration happens faster. So at higher temperatures, transpiration is faster. Next thing is increased wind intensity. Strong winds speed up transpiration by removing water vapor from around the leaf, allowing more evaporation to occur. So imagine this is a sidewards view of the leaf, and so the leaf is full of water. Actually, I'll just do it I'll do it here. So there's the leaf. It's full of water. Water has now diffused out as well. Now, because you've got water on the outside, you do not have as much of a diffusion. You don't have as much of a diffusion pathway here, because diffusion happens where water moves from a region of high. Now, this is the gas we're talking about, which is why I'm saying diffusion and not osmosis. So when we say water evaporates, it's moving from a high concentration to a low concentration, as you would expect in diffusion. So some water is moving out, but not massive amounts because there's water there. But if it's windy, what will the wind do? It'll blow all of these air particles away. Well, not the leaf, but it blows all that water away. Now what we've got is we've got more of a concentration difference. So the water diffuses out. So we have more evaporation occurring. If more water leaves through the leaves, you've got more water then going from the roots up the stem. So that increases the speed. High humidity. Now, humidity, what we don't understand here is what do we mean by humidity? Humidity is how much water is in the air. So humidity is the amount of water in the air. So when humidity is high, transpiration slows down because air already has high moisture content. So again, let's use our leaf. We've got lots of water on the inside, but now we're saying there's lots of water in the air surrounding the leaf as well. So now we don't have as much of a concentration gradient, meaning not as much water is going to diffuse out. Less water diffuses out. That means you've got the transpiration stream slows down. And then finally, we've got greater light intensity. Now, the more light intensity we have, that increases the rate of transpiration because it causes stomata to open. So think back, stomata, the guard cells open. When there's a greater light intensity, more water will enter the stomata guard cells. And when the guard cells become turgid, they become this kind of moon shape. So that's when the stomata opens. So when the light intensity increases, the rate of transpiration also increases because you've got water now going into the guard cells and the stomata is open. The more stomata that open, the more water that will diffuse out, the more evaporation we have. So this needs to be memorized. I've not seen this as a 6-mark question, but this would make an excellent 6-mark question. So how can the rate of transpiration be calculated? So really simple. You get a leaf, you measure the mass of the leaf, Then you suspend the leaf from a string. So you suspend it from a string. After a set time, you remeasure the mass and find the change in mass and divide by the time taken. Because look what we're trying to find out, the rate. Now think back to what I said to you when we did chemistry. When we say rate of anything, what do we mean by rate? Rate means the mass of something or the volume of something divided by the time. So it might be the mass divided by the time or the volume divided by the time. The change in mass, the change in volume divided by time. So here we're finding out the change in mass and we're dividing by the time taken. So that way we know what the rate of transpiration is. Because in this case, either the water has entered or water has exited. Now we're looking more for when water exits. So we're seeing what the change in mass is. Divide that by time and we therefore know the speed at which that occurred, the rate at which it occurred. Okay, now 14.6.14, 6.15, 6.16 are all triple only topics. And that's only 10 slides. So it won't take too long. So for those who are doing combined, so those that are doing combined, it's just gonna be 10 minutes that you'll have to be patient. So surviving in extremes. What adaptations do plants living in hot and dry climates have? So again, this can come up in triple award. This can come up as a 6-mark question. So it's important to have an understanding of different types of adaptations. The 6-mark questions or the questions that have come up on this topic in the past do guide the learning quite well. So it is important to kind of look for these kind of questions linked to this topic within past papers because a lot of it is understanding the context of the question because you're not going to get given necessarily the same plant twice. So number one, it's got a thick waxy cuticle to reduce water loss by transpiration. So usually leaves do already have a thick waxy layer on the top, but in plants that are in really hot, dry countries, dry climates areas, they have a tendency to have a thicker cuticle than usual. The stem that can swell, so it can store lots of water. So it means it'll have more cells and a stronger cell wall. So more water can enter without it bursting. Then you've got long, deep roots. Now, sometimes you've got long, shallow roots or long, deep roots. So shallow roots mean that water can be taken from the surface, particularly after it rains, whereas deep roots go deep into the ground to get hold of that water, and fewer leaves with small surface area to reduce water loss by transpiration. And in many cases where you've got these spikes, they're actually the leaves, but rolled up very tightly, and that stops water loss, reduces the amount of water that's lost. Now with topic 6, there's no required practical, but there are still some practice. There are some practicals that can still be linked to this topic. For example, and actually it's linked to, so for example, investigating the effect of carbon dioxide concentration or temperature on the rate of photosynthesis. So things like this, but they're not required practicals. So there's the core practical link to this topic is, apologies, let me say all that again. So the core practical is the one that we went through that was linked to photosynthesis. But other than that one core practical, there's no others linked to topic 6. Okay, 6.15b, plant hormones. Again, very common question that comes up quite a bit in the triple paper. Generally speaking, for those who are doing triple award, your mindset should be to make sure you know the additional triple content inside out, because you're more like you already know that 30-40% of your paper is going to be triple award. Okay, so plants produce hormones to help manage and guide their growth as well as their reaction to light and gravity. So what is phototropism and gravitropism? Now, gravitropism is also known as geotropism, which is an irrelevant point at this point. Now, phototropism is the process by which an organism grows in response to light. So generally speaking, if it's a sunny day, then a plant will grow towards the light. That's phototropism. So even if it was going this direction, it starts growing towards the light. That's phototropism, the fact that it changes direction. Then gravitropism, this is growth of a plant in reaction to gravity. So where shoots grow upwards, so shoots grow in the opposite direction of gravity, and the roots grow downwards in the direction of gravity. So how does that happen? So this is through the release of a hormone called auxin. So unequal amounts of auxin lead to different growth rates. in plant roots and shoots. So in shoots, auxin promotes growth. So this is what actually happens. You've got light coming from this side. The auxin always goes to the shaded side of the plant. So the auxin moves to the shaded side of the plant. And what auxin does is it encourages cell growth. So that means this part here grows faster than the inside part. In other words, the plant starts growing towards the light. So look, auxin moves to the shaded side of the plant, encouraging cell growth, which causes the plant to bend towards the light. Okay, so I hope that makes sense. We've got auxin here. Auxin causes that side of the plant to grow faster than the other side of the plant. So it means it bends towards the light. Now in roots, it's the other way around. The auxin slows down the growth and it moves towards the side closest to gravity. So the cells grow less on the underside while they grow more growth occurs on the side of the roots away from gravity. So what that means is here's your roots. The auxin builds up here and that actually slows down the growth on the underside. That means you got more growth. Oops. You got more growth on this bit here. Actually, I've done it green one in the roots. Sorry, let me change the color again. It's fine. Let's just do the purple. So you got more growth on the top than you have at the bottom. So the roots grow downwards. So what we're saying this time is the auxin goes to the lower side. So let me just show that again. So let's put the auxin with red. Auxin's on the lower side, which slows it down. So auxin has the opposite effect on the roots. It slows growth down. This top part, the top part grows faster, so the roots bend downwards. They bend towards in the direction of gravity. So we should also know the different uses of hormones. So they can be used to promote growth in tissue cultures. We can use them as weed killers and rooting powders. So rooting powders means you can either take small cuttings from trees, and the rooting powder helps them grow. And similar thing can be done here. Or it can just be when you've got seeds and it helps it grow, that helps the seeds germinate and grow faster. So another example is ethene. It's used in the food industry to manage how fruit ripens while being stored and transported. So what that means is you can take some fruit, for example, an avocado, and avocados come from some have to travel quite far before they, for example, get to the UK. So what they'll do is they will initially, sorry, excuse me, just one second. So what they'll do initially is they'll take them when they're quite unripe. And then just before they go on the supermarket, get sent to the supermarket, they release ethene and then that fruit ripens really quickly. When it's at the right shade of green, that's when they get sold and sent to the supermarkets. Then you can use something like gibberellin, which is another hormone, which ends seed dormancy. So some seeds you can keep in a packet for a long, long time. And then so to increase the speed at which it germinates and starts to turn into a plant, you can add gibberellins, which means they germinate faster, even in conditions where they would not normally grow. So gibberellins help grow plants in areas where the conditions might not be ideal. They also promote flowering, so which is particularly useful in horticulture to ensure plants bloom at specific times. So for example, for people who have show gardens, etc., they would use it at certain times of the year to make different plants bloom. Now on the next slide, we've got a 6-mark question which ties everything together. So auxin is a plant hormone that controls growth. It helps roots grow down and shoots grow towards light. Weed killer is a chemical that uses plant hormones to kill unwanted plants by speeding up their growth too much. Rooting powder is a powder with hormones like auxin that helps plant cuttings grow roots quickly. Just a quick point, whatever you see that capital B, it means it's triple. So rooting powder is a powder with hormones like auxin that help plant cuttings grow roots quicker. Okay, so this is a really good 6-mark question and the mark scheme answer that goes with it. This is the kind of question I would advise that you memorize. So explain the use of auxins, gibberellins, and ethene in the commercial production of plants and fruits. So auxin is used in selective weed killers. It targets broadleaf plants. So these are things that I've not put into the notes because it's all in this 6-mark question. It kills the weeds, but it does not narrow leaf crops. So it attacks the weeds, but not anything else. But no. Auxin is added to rooting powder and it's also added to the end of cuttings to stimulate growth of roots. And then now, generally speaking, when you're answering a 6-mark question, you do not need every single bullet point. But when you're memorizing and you're getting ready for an exam, you would memorize every single bullet point just in case, for example, you get a 3-mark question linked to auxins or gibberellins or ethene. It means you've got all those points there to get your marks. Whatever comes up as a 6-mark question one year is very likely to be recycled over many years as 1-mark, 2-mark, 3-mark questions. So the best thing is to memorize it. Now, overall, in a 6-mark question, you need a few bullet points from each section. Because the question is talking about auxins, gibberellins, and ethene, you need to explain the uses of all three in the commercial production of plants and fruits. So you need to have fulfilled that answer for auxins, gibberellins, and ethene. So you would still need 2 or 3 bullet points per point. Now, gibberellins, they stimulate germination in dormant seeds. They initiate the breakdown of starch. So this is what they do inside. They cause the starch to break down. Now, when starch gets broken down, it gets turned to glucose. Glucose is then used in respiration. That energy that's released is what helps the plant grow faster. That stimulates flower formation. It promotes fruit formation because if the starch is broken down to glucose, then that glucose is going to need to be stored somewhere and it's stored inside the fruits. It's sprayed onto plants before pollination and it stimulates development of seedless fruits. Okay, ethene is used for fruit ripening. So initially the unripe fruit is harvested and it's like I said before, and just at the right time, ethene is released, and then because the ethene is released at the just the right time, it means the fruit doesn't go bad on the journey to wherever it's going. So if you've got avocados growing in, for example, South America, and then they put on a big ship container and they make their way to the UK. You can pick them off the tree unripe, and you can add the ethene just as it's coming, just before it's sold. So it ripens at just the right time. That way it doesn't go off. Okay, so let's continue. And still it's biology, Edexcel paper 2, and we're looking now at topic 7, animal coordination, control, and homeostasis. Now in paper 1, we looked at the nervous system and we looked at those fast responses to things. Now what we're looking at here is the endocrine system, which is made up of glands, and it's glands that secrete hormones directly into the blood. So that means hormones travel at the speed of the blood. The blood carries the hormone to a target organ, which leads to an effect. However, although hormones target specific organs, it can still have a knock-on effect on other parts of the body. So the endocrine system is slower and longer acting than the nervous system. So these are some of our important glands. So we've got the hypothalamus, that's linked to temperature control. The pituitary gland is the most important one. That's the one that we call the master gland. It's the one that sends hormones to, for example, the thyroid gland, and then the thyroid gland releases a different hormone. It sends it to every single one. Adrenal gland releases adrenaline. Thyroid gland releases thyroxine, which is linked to our metabolism and how fast we digest food and do different chemical reactions in the body. The pancreas releases insulin and glucagon, and then we've got the testes that releases testosterone. Testosterone promotes the production of sperm cells. Ovaries cause the release of estrogen. Estrogen promotes the release of egg cells. So each one here is quite important. And like I said, hypothalamus. The thymus is linked to the thyroid gland. So I'm not going to go into too much detail with that. But we need to know every single one. Particularly, hypothalamus is more of a triple element there. But the remaining will come into combined as well. So the pituitary gland is located in the brain. It's known as the master gland. It releases various hormones into the bloodstream. Those hormones influence other glands to secrete additional hormones. So here's the pituitary gland. When it releases ADH, that causes the kidney that has an impact on the kidney. When it releases TSH, the thyroid gland releases thyroxine, or it reduces the amount of thyroxine it releases. When it releases FSH-LH, that causes the testes to release testosterone. So the pituitary gland causes other glands to secrete additional hormones. Okay, so let's start with adrenaline. Now, adrenaline is released by the adrenal gland, right by the kidneys. Adrenaline readies the body for fight or flight. Now, there's a reason why. So whenever we have moments of fear or stress, the adrenal gland releases Adrenaline. Now, what the adrenaline does is it increases the heart rate. Now, when the heart rate increases, it increases the supply of oxygen. So you can imagine if the heart's beating faster, it's beating faster. So if it's beating faster, what's the significance of that? The significance of it beating faster is that it's going to pump more blood around the body. What's being pumped around the body that's really important? Oxygen and glucose. Why do we need oxygen and glucose? For respiration, to release energy, particularly to the heart and the brain. So that means we've got more energy to deal with the situation. So if we're in a situation where we're scared or we're stressed, our body needs to either be able to fight or run away. So if we're in physical danger, this hormone is released and it either increases the speed at which we run away or it gives us that little bit of an extra boost to fight. So adrenaline prepares our body for stressful and fearful moments. Thyroxine. Now, thyroxine is released by the thyroid gland, so secreted by the thyroid gland. Thyroxine is the hormone and it's secreted by the thyroid gland, and this boosts the body's basal metabolic rate, as in it boosts our rates of different reactions occurring in the body. So thyroxine plays a crucial role in growth and development. So its production is regulated by a negative feedback mechanism. So this is a higher only topic. It will not come on the foundation paper. Now, negative feedback is also a higher only topic. It's important that we understand what that means. And I'm gonna show you now. Okay, so it says here low levels of thyroxine. So let's start with that. We have low levels of thyroxine. So let's call it T. Thyroxine is T. Now, that triggers the hypothalamus to produce TRH. So we get an increase in TRH. That causes the pituitary gland to release TSH. So we now have an increase in TSH. Why is that significant? Because TSH acts on the thyroid gland. And what does the thyroid gland do? It produces thyroxine. So because of our increase in TSH, we now have an increase in thyroxine. Now because we have an increase in thyroxine, it prevents the release of TRH. So now we have a decrease in TRH. Now, if we've got a decrease in TRH, then the TSH production gets stopped. Now, we just said TSH. What did TSH do? It caused an increase in thyroxine. Now, if TSH has decreased, that means thyroxine is no longer made. And look, thyroxine levels start to decrease now. If thyroxine levels decrease, TRH is produced by the hypothalamus and it increases. If TRH is made, once that hormone reaches the pituitary gland, there's an increase in the production of TSH. What does TSH do? It increases the amount of thyroxine made. So if you increase the amount of thyroxine made, that decreases there. So now we've increased the thyroxine. If thyroxine levels go up, the TRH levels decrease. If the TRH levels decrease, the TSH levels decrease. What does that mean? Less thyroxine is produced. So now we've got low levels of thyroxine again. So we're back to having low levels of thyroxine. If we've got low levels of thyroxine, the hypothalamus starts to produce TRH. When that hormone reaches the pituitary gland, TSH starts to be made. An increase in TSH, what does that do? It increases the amount of thyroxine. So thyroxine has increased. What does thyroxine increasing do? It reduces the amount of TRH, which reduces the amount of TSH, reduces the amount of thyroxine. So we can just keep going around in circles over and over again. However, I'll stop at that point. You can, if you don't understand, just watch. Now, if you didn't understand, then just watch these last 2-3 minutes on loop until it does make sense. Because what we're saying is when thyroxine levels go up, the body finds a way to reduce them. If they go too low, the body finds a way to increase them. And by doing that, our body roughly keeps the same amount of thyroxine within the body. Because it's one of the homeostasis things. It's something that needs to be kept constant in the body. So thyroxine levels are controlled by negative feedback. And how do you know it's controlled by negative feedback? Anything where if it goes too high, it's brought back down. If it goes too low, it's brought back up. That's negative feedback. So if our body's temperature goes too high, our temperature increases, our body deals with that and it cools our body down. But if our body temperature goes too low, then our body deals with that and it does whatever's necessary to bring the temperature back up. So there's a few things that our body maintains and thyroxine is one of those. So I've already explained this, a mechanism that balances levels by decreasing them. when they're too high and increasing them when they're too low. That's what negative feedback is. And this I've explained as well on that previous slide. Okay, menstrual cycles. This is something that we need to know for if it's higher, foundation, combined, triple. Everyone needs to know this. Now, estrogen is one of the main female reproductive hormones produced in the ovary. So the ovary is the gland. Estrogen is the hormone. And because of estrogen, eggs start to mature and it leads to ovulation. Now, in males, testes are the gland. Testosterone is the hormone that's released by testes, and that leads to sperm production. Right now, this, if we can understand this logically, then it makes the whole of this topic really easy. If you learn this one slide, 6-mark question, 3-mark question, whatever it is, it'll make your answering of questions much easier. And I'd advise lots and lots of past paper practice. I've not seen a single paper where this hasn't come up. Now, don't get me wrong, there's been a few topics that have come up every year, but haven't come up this year. For example, enzymes wasn't covered too much on the previous topic. However, don't forget that enzymes is B1, and B1 can come up in so it can come in either paper. So alongside revising this and doing lots of past paper practice, don't forget to revise your B1 stuff. Now, B1 and C1 are both part of your paper 2 exams as well, and you can recap them on the Learning YouTube channel from AMALathon2. So if you go to the live section, B1 and C1 are covered. Right, so let's go through this one at a time first. Let's look at FSH. What does FSH do? It causes an egg to mature in the ovary. So in the ovaries, and I'm about to draw some really bad diagrams, but hopefully it still makes sense. So there's the ovary, there's the oviduct, and there's the uterus. So FSH causes the egg to mature in the ovary. Now, in the actual ovary, there's lots of eggs, but they're not mature, meaning they can't be fertilized. So FSH causes them to become mature in the ovary. And that stimulates the ovaries to make estrogen, which is another hormone. So now let's go to estrogen. Estrogen causes the lining of the uterus to build up. So there's the lining of the uterus becomes thicker. Now that's really important because the normal uterus is not thick enough for the fertilized egg to embed into. The uterus lining has to build up so that the fertilized egg can implant and start, and so that the pregnancy can start. Once the fertilized egg, so the egg and the sperm have joined, the fertilized egg embeds itself into uterus, it starts to multiply. And that's where the placenta is formed. That's where the baby starts to produce from. Now, what the estrogen does is it stops FSH. And that's important because we don't want another egg to be released. So what did FSH do? It caused one of the eggs to mature. And there's the egg, matured and ready. And it also caused estrogen to be produced. What did estrogen do? It caused the uterus line to thicken, to build up, so that it would be ready for the implantation of the fertilized egg. Now, estrogen also stops FSH, which is important because we don't want more than one egg being released, but it starts LH release. So luteinizing hormone, that causes the egg to be released. So now the egg is released, it goes down the fallopian tube or the ovarian tube and enters the uterus. Now, if it fertilizes, it will then embed itself into this thicker layer of the uterus. Now, at this point, after LH, that stimulates the release of progesterone. The progesterone maintains the uterus lining, so it maintains it. And also it stops LH and FSH. Now, that means it inhibits its production. Why is that important? Because it stops FSH because we don't want another egg to be matured. And it stops LH because if for whatever reason the egg did become mature, we don't want the egg to be released. At this point, that one egg's already been released. So now, if a viable pregnancy has occurred, progesterone levels start decreasing. That means this uterus lining starts to break down and it's released from the body as menstruation. At this point, FSH is released again, and that means that a new egg starts to mature and the new cycle starts. So let's go through that one more time. I hope everyone can see the logic, how one feeds into the other. So again, let's draw. Okay, so FSH, it causes an egg to mature in the ovary. So here's the ovary, and there's our mature egg. It stimulates the ovaries to make estrogen. So what does estrogen do? It causes the uterus line to build up. Why do we need the uterus lining to build up? So that the fertilized egg can implant into the uterus lining. It stops FSH because we only want one egg in there. We don't want multiple eggs, but it starts LH release. So the luteinizing hormone, or LH, it causes the egg to be released, ovulation. So now the egg's been released and it's entered the uterus. If it, at this point, if the egg cell is fertilized by a sperm cell, it will then implant itself into the uterus line. It's not going to be quite that big, but I've just so you can see what I'm talking about. Now, LH causes the release of progesterone. What does progesterone do? It maintains the uterus lining. So it maintains it. And if it's a viable pregnancy, as in the egg cells been fertilized, then what will happen is progesterone will maintain this layer, this uterus lining for the whole of the pregnancy, and then the baby will develop in the womb. Now, if it's not a pregnancy, the sperm cell has not sorry, one thing to remember here is this stops LH, that means no more eggs will be released, and it stops FSH, which means no more eggs will mature, which if it's a pregnancy, neither of those things are needed. Now, as soon as if it's not a pregnancy, the progesterone levels start decreasing, which means FSH starts to be produced. So another egg is matured and the whole cycle starts again. And the uterus lining breaks down and the menstrual cycle starts, as in the period starts. Right, so this is exactly what's being said here. So FSH, oh sorry, just one thing I should point out. Now, for chemical hormones, for chemical hormone-based contraception, sorry. So you've got non-hormonal contraception, like condoms and cream, certain types of creams, etc. And then you've got hormonal contraception. So one of the main things in hormonal contraception is progesterone. Why? And it makes sense if you think about what progesterone is. What does progesterone do? It stops LH and FSH. And you've got to think, why is that significant? Because what does FSH do? It matures an egg. So if the progesterone stops FSH, it means an egg will not mature. If the egg hasn't matured, a sperm cell can't fertilize it. And then you've got LH, which causes the egg to be released. If the egg isn't released, it's not going to be fertilized. So progesterone is one of the main things inside hormonal contraception, as in that's what's inside the pill for ladies that are trying to not become pregnant. And then for those who are trying for pregnancy but they're struggling, they take tablets with FSH and LH because FSH will mature the egg, LH will ensure that it gets released. So FSH is released, that causes the egg to mature and estrogen to be released from the ovaries. So now we've got estrogen. What does estrogen do? It causes the lining of the uterus to build up. It stops FSH, which is why estrogen is also one of the hormonal contraceptive, one of the contraceptive hormones as well, because it stops FSH, but it does stimulate LH release. So now the egg's matured, it's been released, and there's a thick uterus layer. LH causes the egg to be released, and then progesterone prepares the uterus lining for implantation of an egg. And at the same time, it inhibits LH and FSH. FSH, it stops FSH from producing, maturing another egg, and inhibits the release of LH. LH is the hormone that causes the eggs to be released. Now, as progesterone levels fall, the thickened uterus will break down and it's discharged during menstruation. Now, as the progesterone levels decrease, FSH and LH levels start increasing again. So the whole cycle, the whole process restarts. Okay, so how can fertility be controlled? Again, this is something that can easily be turned into a 6-mark question, so make sure you know your differences. So an oral contraceptive pill, it's hormonal. It contains progesterone and sometimes estrogen. That stops FSH, so that way the eggs don't mature. Now, advantage is easy to take. It's highly effective. It definitely stops pregnancies, but it can cause side effects and it has to be taken daily. It can lead to high blood pressure. It will not prevent sexually transmitted infections because it's not a barrier to the sexual fluids. Then you've got barrier methods, for example, condoms, etc. They're non-hormonal. They prevent sperm from entering the uterus, and they have no side effects unless someone is allergic to the material. And they prevent STIs. They can break or tear though. So there is a disadvantage. Then you've got this injection. It's hormonal. It slowly releases progesterone to inhibit the maturing and release of eggs for a number of months. So once this is injected into a person's body, it just it stays there. So meaning you don't have to take a pill every single day. So that's an advantage, but it does have lots of side effects. No one's body should have 3 or 4 months of additional progesterone for no reason. So it can have lots of side effects. But it means for for someone who doesn't want to take a pill every day, it's that does have that advantage. Okay, so spermicidal agents, they're non-hormonal, they kill or disable the sperm. They can be used alongside the barrier contraceptive. So this is just a cream that kills sperm. So it can be either put inside the vagina or over the barrier method of contraception. But the issue here, it can cause allergic reactions. Surgical methods, they're non-hormonal. So what does it do? It's sterilization. In other words, the tubes that carry the eggs or the sperm can be cut. That means the egg won't be released or sperm won't be released. So this is very effective at preventing pregnancies, but it is difficult to reverse. Okay, so 7.8, this is higher topic, it's assisting reproduction. So what is assisted reproductive technology? So initially, for this is, for example, someone that's struggling to have a child. So it could be IVF or the fertility drug clomiphene. So this is the use of hormones and procedures to help achieve pregnancy. Now, hormones can be used to help treat infertility. So a patient could take FSH and LH because FSH matures the egg. LH will then cause the egg to be released. And what that means is if a female does take FSH and LH within medicine, she might become pregnant the normal way. It could just be that her body doesn't produce enough of those two hormones, which means eggs aren't being matured and released every single month. So taking this just guarantees that there's that release of the hormones, the release of the egg. Now, IVF is where the egg cell is fertilized external to the body. So, for example, the mother receives FSH and LH that promotes the maturation of multiple eggs. So now this lady that wants to become that's struggling to become pregnant takes these two hormones. She's released loads of eggs. Now what the doctor will do is take those eggs out of the mother and then out of the body fertilize it with sperm. So now this part's happening inside the body. This part onwards is outside of the body. So the egg cell has been removed, sperm has been taken from the father, and they've been mixed together. So the egg cell becomes fertilized by the sperm cells. At this point, the eggs are then fertilized with the father's sperm in a lab. The fertilized eggs develop into embryos. That takes about 6 days, 7 days. And once the embryos, now when we say embryos, we're talking about when they're just a few cells. We take small cluster cells, and they take 1 or 2 embryos, and they're placed into the mother's uterus. So they are then placed into the mother's uterus, and that can lead to a successful pregnancy. Now, the only issue is that this is emotionally and physically stressful. Success rates are not high. It is almost like a last chance. Hopefully, this will work kind of thing. And it can also lead to multiple births because if you look at this part here, they put 1 or 2 embryos. So sometimes they put a few embryos in just to make sure at least one of them embeds itself into the uterus lining. And sometimes all 2 or 3 of them embed. They end up having twins or triplets. Non-identical twins and non-identical triplets, but still twins or triplets. Now, having multiple births can cause a risk either to the babies or to the mother. So what's clomiphene therapy? So clomiphene is a drug. It's given to women who are not ovulating, and that can be due to a blockage that the egg's not being released. So the drug blocks the action of estrogen, which inhibits LH. So this clomiphene therapy stops estrogen. That means LH, so that means there's going to be a massive amount of LH, and that means an egg is more likely to be released. Because what's the whole point of LH? It stimulates the release of eggs. So by blocking estrogen in the body, there's going to be more LH because estrogen inhibits LH. Okay, so we're going to continue from 7.9. What is homeostasis? So homeostasis is the regulation of a cell or organism's internal environment to ensure optimal conditions for functioning. What that means is the body's attempt to keep temperature the same, to keep the glucose concentration the same, to keep water levels the same, even keeping certain hormone levels the same as well. So that's homeostasis, is the body controlling certain things. Now, this controlling of blood glucose is for everyone, but to understand how water levels are controlled and body temperature is controlled, that is triple. So there are elements of this lesson that will be linked to triple. So why is it important that these are controlled? So one thing that stands out straight away is body temperature and enzymes. So cells, enzymes, and life processes function only in certain conditions. So our body does its best to keep our bodies at optimal within a narrow range and at optimum. So keep it at optimal conditions. So conditions in the body are controlled by homeostasis include water levels, blood glucose, and body temperature. But a good example of why it's important, body temperature, anything beyond 37 degrees, and what starts to happen to enzymes, they start denaturing. When we go below that temperature, then the enzymes don't work as quickly. So these automatic control systems can include either nervous or chemical responses. All control systems consist of. So our body's control systems are made up of receptors, coordination center, and effectors. So this is, you would have seen when we did nervous system. So receptor includes things like the skin, which detects temperature and pressure. It detects changes in the environment, stimulus, like a change in temperature. It then sends a message to the brain or the spinal cord. The brain and spinal cord receive that information from the receptor and they pass a message on to the effector. Now, the effector can either be a muscle, and that's an example of a nervous response, or it can be the glands, which is an example of a chemical response. So our body can react to a scenario, a situation, a stimulus, a change in the environment in both of these ways. One with a quick response and one with a slow response through the glands. So the muscles, when they get a nervous impulse reaches them, they contract. When a nervous impulse reaches the glands, they secrete chemicals. And sometimes that gland, as we already learned, can cause another gland to secrete enzymes as well, secrete chemicals, hormones. Okay, 7.10b, and that means this is triple only. 10, 11, and 12 are all three are triple, and then we're back to combining triple again. So 7.13 onwards will be for everyone. Now, why does the body temperature need to be controlled? As we just mentioned before, if the temperature is too low, then we've got a low rate of reaction because the enzymes don't work well enough. They don't move, they don't have as much energy, they don't move around as quickly. You have less collisions. Less collisions means less reaction. And at high temperature, the enzymes begin to denature. And whenever the word denature comes in, one thing we've got to remember here is that you need to know topic one inside out. Topic one is part of your paper 2 exam. So if anyone's forgot topic one, if you go to the Learning YouTube channel, you can watch and go through all of topic one. Just like I'm doing topic 7 right now, I've covered all of topic 1 as well in the live section. So if an enzyme denatures, what happens? The active site changes shape. If the active site changes shape, it means the substrate will no longer fit, and substrate no longer fits. What does that mean? It means there's no chemical reaction. So whenever you talk about enzymes denaturing, you've got to go back to those basics. Okay, so what happens if cells are in dilute solution, such as dilute blood? So increased blood water levels. So if there's too much water in the blood becomes more diluted. Water enters the cells via osmosis. Consequently, this can lead to cell swelling and the risk of bursting. Now, just to clarify here, If you've got more water on the outside, then the water is going to enter the cell. But when we have too much water, as you're going to find out afterwards in this lesson, certain hormones are released and more water enters. The kidneys become more permeable. The tubules become more permeable, or sorry, less permeable, and more urine is made. And that means more water is released in urine. So when we drink lots of water, it's a healthy thing. It's not a problem. This is extreme amounts of water. And people drink extreme amounts of water usually when they've taken certain drugs. And by drugs, I mean recreational, not medicinal. So drinking 2, 3, 4 liters of water is not going to cause this problem. Now, what happens if the blood is low, has low water levels or in a concentrated blood? Then most of the water is going to come straight out. And that is a problem. And that can lead to the cells shrinking and not functioning properly. So if someone wants to have good skin, better than any cream, drink lots of water. Okay, 7.11b, thermal regulation. So it's how the body keeps our temperature at 37 degrees. So the thermoregulatory center in the brain is responsible for monitoring and regulating body temperature. The center is equipped with receptors. So within the thermoregulatory center, which is in the brain, it's got receptors that are sensitive to the blood's temperature. So if the blood's temperature is too high, then we do this. We start sweating. So sweat glands generate perspiration, meaning we sweat. The blood vessels expand, in other words, vasodilation. And we also, our hairs lie flat. Now, when our hairs stand up like this, then air gets trapped in between. And air is a very good insulator, so it keeps the heat in. In this case, the hairs lie flat. The hairs lie flat because less air is trapped, so more heat is lost from the skin. Now, when sweat is produced, what happens is, so imagine this is someone's arm, it's covered in sweat. Now, the heat that's inside the body is used to evaporate that sweat. So that means the heat energy goes into the water, into the sweat. And that means the sweat absorbs that heat energy and takes the heat energy out of the body. And when it evaporates, it takes that energy with it. So sweat absorbs the heat energy and then it uses that heat energy to evaporate. So it cools the inside of the body up. So it cools it up. Cools it down, so it cools up. Cools it down, sorry. It cools the body down. Blood vessels widen, so that's vasodilation. That just means more blood vessels are near the skin, so more thermal energy is lost to the air. And this goes alongside with the sweat. Because the blood vessels are closer to the skin, that means there's more heat energy towards the surface that will heat the water up in the sweat. Now, during physical activity, the body requires additional energy. Now, whenever we need energy, what should automatically come to mind? Again, from topic 1, mitochondria. What happens inside the mitochondria? Respiration. And what does respiration release? Energy. As respiration occurs, heat is released. And that means the body's temperature rises. So this is why sometimes it's important to reduce that heat energy straight away to help that heat energy escape as quickly as possible. That's why we sweat when we run. Now, what happens if the body temperature is too low? Oops, sorry. What happens when the body temperature is too low? Sweat glands stop making sweat because we don't need to lose any heat energy. Nor do we need water on our skin that freezes. Blood vessels narrow, so vasoconstrictions. That means the heat energy it goes towards the center, towards the inner part of the body. So the heat energy that escapes within the body, and we get skeletal muscles contracting, resulting in shivering. Now, again, for them to contract, what does that mean? Shivering stimulates increased respiration. So muscles have to contract and relax. That means we need more mitochondria, lots of respiration, releasing energy, but this time the energy that's released warms the body up. So not producing sweat. Vasoconstriction, it reduces the energy transfer from the skin to the surrounding environment. So this is a summary of what happens when it's a cold day or the temperature is too low. Hairs stand up because the hairs that have stood up, like I explained before, they trap more air in between the hairs, and that acts as an insulating layer, keeping the heat in. No sweat is produced. That means there's no sweat there, so it won't be heated up by the body, causing evaporation. So it removes that process. Blood vessels narrow, vasoconstriction. Less blood flow flows near the skin, so less heat is lost. And shivering. Muscles contract quickly, which makes heat from respiration. So if it's too hot, now this is an example of negative feedback. When it's too hot, we have vasodilation, sweating, hairs lie flat. So our temperature goes back to normal. But when it's too cold, now that means our temperature starts decreasing. Now if the temperature goes too low, then vasoconstriction happens. We start shivering, hairs stand up. Then the temperature starts to increase until it gets to normal. At some points, it can go too hot. So our body cools it down. When it goes too cold, our body warms it up. So our body works a little bit like a thermostat. So a thermostat in the house, what it does is, if for example you set it at 20 degrees, if it goes slightly above, the radiators get turned off. If it goes below, the radiators get turned back on. If it goes above, radiators get turned off. If it goes below, radiators get turned back on. So it maintains that 20-degree temperature. And that's what our body's doing. It's maintaining that 37-degree temperature by, if it becomes too hot, it very quickly cools the body down. If it becomes too cold, it quickly warms the body back up. Okay, controlling blood glucose. So controlling blood glucose is, we're back to combined, triple, everyone. So those that just took a small break, you can come back now. This is for everybody. So how are blood glucose levels controlled? So this is for combined, triple, everyone. So hypoglycemia, when we don't have enough sugar in our blood, and hyperglycemia, when we've got too much sugar in our blood. Both can cause illness. Both are important to understand. So how are blood glucose levels controlled? So blood glucose concentration is monitored and controlled by the pancreas. Now, if the pancreas detects that blood glucose concentration is too high, so the pancreas detects that blood glucose is too high. So what does it do? It releases a hormone called insulin. Now, the insulin is picked up by the liver and the muscles, and what the liver and muscles do, because they have receptors inside them, the receptors attach to the insulin and they start absorbing glucose from the bloodstream. And what the liver and the muscles do is they convert glucose into glycogen, this word here. And then that glycogen is stored inside the muscles and inside the liver. Now, if the blood sugar level goes too low, what I'm about to go through now only comes up in higher. So blood glucose going too high and insulin being released by the pancreas, and then the liver and the muscles converting glucose to glycogen, that's for higher and foundation. But if the blood glucose level goes too low, what happens next is all part of the higher paper. So now this time the pancreas releases a different hormone called glucagon. Glucagon, so this is supposed to be the pancreas, so I've drawn the line in the wrong place. Glucagon is picked up by the muscles and the liver, but this time what the muscle and liver do is they convert the glycogen back into glucose, and they put all that glucose into the bloodstream. So what happens? The blood glucose starts to increase. Now, if the blood glucose becomes too high, if the blood glucose becomes too high, then this happens again. The pancreas secretes insulin. Insulin is absorbed by the liver and the muscles. They convert glucose to glycogen. That takes glucose out of the bloodstream. So the blood glucose decreases. If it becomes too low, then we go through this process again. Glucagon, the hormone glucagon, is released by the pancreas. Glucagon is picked up, there's receptors in the liver and the muscles that attach to glucagon. The liver and the muscles start converting glycogen back into glucose. Glucose then goes into the bloodstream and the blood glucose levels increase. So we're back to here again. When the blood glucose levels go too high, the pancreas secretes insulin. There's receptors in the liver and the muscles that attach to the insulin. They then start converting the excess glucose in the bloodstream and converting it to glycogen for storage. And it's this cycle goes on and on and on. This is another example of negative feedback. If the blood glucose levels go too high, insulin is released and the blood glucose levels come back to normal. If they go too low, glucagon is released and that causes glucose to be put back into the blood. So if the blood glucose levels are too high, insulin is released and the amount of glucose in the blood is decreased. If the blood glucose levels go too low, glucagon is released and that causes more glucose to be produced from the glycogen and put back into the blood. So this is negative feedback, maintaining that one amount of glucose. So 7.15 and 7.16 is linked to diabetes. Now, diabetes is a condition that affects the body's ability to either produce or respond to insulin. So there's two things that we're looking at here. Type 1 diabetes is when a body cannot produce insulin. Sorry, type 1 diabetes is when the body cannot produce insulin. Type 2 diabetes is when the body cannot respond to insulin properly. Now, so type 1 diabetes, it cannot produce insulin because there's a problem in the pancreas. This is an inherited disorder and it's treated with insulin injections. Type 2, on the other hand, it cannot respond to insulin properly. It's linked to obesity and poor diet, and it can be treated with lifestyle changes and medication. So type 1 diabetes doesn't produce insulin. Type 2 diabetes cannot respond. It's where the body stops responding to insulin. So that means in type 1 diabetes, if you check the blood, you'll have very low levels of insulin. In type 2 diabetes, the body is still making normal amounts or excessive amounts of insulin. So it's making lots and lots of insulin, but what's the problem? The body is just not responding to it. Okay, so type 1 diabetes is treated with insulin. Now, it has to be an insulin injection. And why does it have to be an injection? Because insulin is a protein. So if you were to eat it, as soon as it enters the stomach, the protease enzymes in the stomach would break it down. So it won't have an effect. So for that reason, it has to be injected. And it is passed on through genetics and family history. So it is purely a genetic thing. It's a recessive allele, so it has to be passed on by both parents. Okay, now this is a really good summary of the differences between type 1 and type 2 diabetes. So type 1, the pancreas can't make insulin. But in type 2, the body doesn't respond to insulin. So if anything, it's producing plenty of insulin. It's just not responding to it. That's why there's there's a lot of scientists, researchers looking at stem cell therapy to replace the cells that are faulty. Risk factors. Type 1 is genetic inherited. By risk factor means what causes it. Now type 2 is linked to being overweight, over 40, high cholesterol, and it can also be linked to genetics. So if someone's got a grandmother or a grandfather or a relative, close relative that's got diabetes, but particularly parents and grandparents, then an indicator that that individual needs to really look after their their weight, their diet before they hit a certain age. Or it just indicates that they're more likely to get type 2 diabetes. Doesn't mean they will get it, but they can get it. Now, treatment and prevention. Type 1 can be treated with insulin injections. Type 2 is controlled by eating healthy, losing weight, and exercise. So type 2 can come under complete control. And it's something that I've come across. People that became diabetic with type 2 diabetes in their 40s, and they spent a couple of years doing a lot of exercising, sorting their diet out, and now the type 2 diabetes is almost not there. It's still there in some respect because it's not going to just go away, but they've controlled it. They're really, really healthy again. So what we can see here is two different ways of tracking blood glucose. Because people with either type 1 or type 2 diabetes have to need to monitor their blood glucose levels. So this one is well, let's start with this one here. What this one does is you press the button and it jabs the finger so that a bit of blood comes out. Then that blood can be added to a device. And by being added to that device, um, by being that what that device does, then it tells you how much sugar is in the blood. This one is a little bit different. This stays attached to the skin for much longer, for a week or so, and it keeps sending messages to a person's phone. So essentially just downloading an app, and it sends messages with how much blood glucose levels. So this is really, really good for monitoring. This is really good for monitoring blood glucose levels, seeing after you've eaten food how much it goes up and how quickly the blood glucose levels come back down again. Now, if we look at these two graphs, and these kind of graphs can come up in exams, I might say, how can you tell the red line? How can we tell that this red line is someone that's diabetic? And there's two things that you can see straight away. Number one, the initial blood glucose levels are much higher. And number two, so this is where a person's eaten food. And as you can see, the initial blood glucose levels are higher anyway, but it also takes longer for them to come back down. So blood glucose levels fall much slower compared to someone without diabetes. As you can see here, the person eats, blood glucose goes up, which is understandable. Insulin is released, so very quickly the blood glucose levels are brought under control again. A diabetic person takes longer. Now, in type 1 diabetes, blood glucose levels are too high, and in type 2, blood glucose levels are too high. But how can we tell the difference between this one and this one? Because this graph represents type 1 and type 2, the red line, when it was red. The top line represents both. But how can we tell the difference between the two? Is it's in insulin levels. So blood glucose is really high, but in type 1, insulin levels are very, very, very low. Because type 1 diabetes means the body doesn't produce enough insulin. But in type 2 diabetes, the insulin levels are normal and sometimes they're really high because the body's trying to compensate for the fact that the insulin is not having an impact. So it keeps making more and more and more of it. So if you're interpreting a graph or a table of results, when you're looking for type 1 diabetes, you're looking for high glucose level. You're looking for the fact that the glucose levels does not drop, so it goes up and it comes down really slowly. Someone without diabetes, it would look like that. But we're looking for that in both high glucose levels, but the glucose level doesn't drop. In type 1, we're looking for low insulin levels. And in type 2, we're looking for high insulin levels. And that becomes an indicator. So now if we look at this information here, you've got person A who's got, this is looking at insulin and how much insulin is in the blood. So here we've got high insulin level. Here we've got low. This is normal range. So this indicates diabetes type 2 because insulin levels are really high. And and 20 indicates type 1 diabetes because not enough insulin has been produced. Okay, BMI, body mass index. This can be used to predict the amount of fat, but this assumes that the mass of other body tissues is in proportion to the height. So this is an indicator of if someone's overweight, obese, or extremely obese. And it's calculated by doing mass in kilograms divided by the height squared. Now, the number of people that forget to square is unbelievable. A lot of students, we're not talking about in just, it might not just be in your own particular school, every school up and down the country. Students forget to square. Same thing happens in physics when they do kinetic energy equals half mass times velocity squared. People forget to square. So instead of getting 3 marks, they sometimes get 1 mark. Now, the issue with BMI is that if you've got someone that looks like this, but they are very muscular, their BMI might actually be somewhere around there between the overweight and the obese category. But in reality, they're just muscle. Muscle is heavier than fat. So BMI is a good indicator, but it's not acceptable or accurate in every circumstance. Now, a lot of the questions that are linked to BMI and waist to hip ratio, questions like this are usually evaluative questions where you're given lots of information and you've got to break down that information. So that is something that when we do the pre-exam workshop, we'll be going through in more detail. But I'm sure before that, you'll have a lot of you, hopefully all of you, will have gone through lots and lots of past papers. The more past papers you do, the better you're going to do in the exam. Okay, this is your understanding phase, which is important that you understand everything. Alongside the understanding, as in the activating of the brain, you've got to memorize and you've got to apply that learning as well. Okay, now the rest of this PowerPoint is linked to triple. So 17.8b, the urinary system. What is the structure of the urinary system? So we've got a renal vein that transports deoxygenated blood away from the kidneys. And we've got the renal artery as well, which transports oxygenated blood to the kidneys. Now, the function of the kidneys, they regulate water content and they filter the blood. So they remove poisons and toxins from the blood, and they also regulate the amount of water. So excess water enters through the kidney into the bladder. So the ureter transports urine from the kidneys to the bladder. The bladder fills with that urine and then it's urinated out. But a lot of the poisons and toxins in our body end up in that urine, and then they're excreted. So the bladder is a store of urine, and then we've got the urethra through which we remove those toxins. Okay, so structure of a nephron. How is excess water, ions, and urea actually removed? So the kidneys remove excess water, ions, and urea through the urine. So all this excess within the blood is lost through the urine. So the kidneys help keep the body's water balance in check. They produce urine by filtering the blood and selectively reabsorbing useful substances like glucose, ions, and water. So initially what the urine does, it acts like a nephron, basically works like a filter. And then any excess, because large molecules don't enter the kidney. So large molecules like proteins, etc., won't enter the kidney. But small molecules like glucose, ions, water, things that dissolve into water will enter the kidney. And then what the body will do, they will actively transport out anything that's useful, like glucose and certain ions, and water will be reabsorbed. So filtration takes place in the glomerulus and the Bowman's capsule. Glucose is selectively reabsorbed and water is also reabsorbed. Now, the bit we really need to understand is this bit, the ADH bit. How is it the ADH helps with the reabsorption of water? So when the blood is too concentrated, meaning there's not much water, the pituitary gland, so the PG, the pituitary gland, secretes ADH. This increases the permeability of the kidney tubules. When we say permeability, it means there's now more space for the water to leave through. Now, because the kidney tubules are more permeable, more water is then reabsorbed. More water re-enters the blood. That means because more waters come out of the kidneys and re-enter the blood, it means the urine that is made will be small, will be a smaller volume. Because what we've just done here is we've taken a lot of the water back so that the blood is not too concentrated. So on a hot day, when it's hot and the person's sweated a lot, a person doesn't have as much water in the blood, the pituitary gland secrete means makes and releases ADH, antidiuretic hormone. The antidiuretic hormone, the effect this hormone has on the kidneys is it makes it more permeable, meaning there's more gaps for the water to go in or out through. Through those gaps, more water is reabsorbed. So that means now the blood is now full, has more water inside it, but it means the urine will be made up of less water. Now, the pituitary gland stops releasing ADH. So what happens if the blood concentration is too dilute? Too dilute means lots of water. And having too much water is a problem as well because it can cause cells to burst. Now, ADH stops being released. So now we've got so the pituitary gland stops releasing ADH, and that causes the tubules in the kidney to become less permeable. As a result, less water is reabsorbed, and that means a large volume of dilute urine is made. So the excess water is urinated out of the body. So you can see it straight away. This is an example of negative feedback. Because one thing increases, one thing decreases. So meaning as when the water is too high, the body stops releasing ADH. So we have a person produces a large volume of urine. So the excess water is released. When the blood concentration, when the blood is too concentrated, as in there's not enough water, then ADH is released. That makes the tubules more permeable, so more water is reabsorbed and a smaller volume of urine is produced. So that increases the blood water levels. So that's negative feedback. When the blood water level goes too high, the body urinates it out. When the blood concentration levels, the water concentrations in blood decrease, so there's less water, then we retain more water and we have a smaller volume of urine. So negative feedback. We have to know these inside out, these examples. Okay, kidney failure. What are the treatments for kidney failure? People who suffer from kidney failure will need a transplant or dialysis. Now, dialysis, the advantage of dialysis is that it keeps the person alive. I'm actually just going to go through this one first. about how dialysis works before going through the advantages and disadvantages. So, blood containing high levels of urea is taken from the vein in the arm. You can see here one vein goes into the machine, sorry, one tube here goes into the machine, and one tube is coming out of the machine. So the tube going into the machine is full of urea. Now, again, let me just quickly explain this. How is urea produced? So when we eat lots and lots of protein, it gets broken down into amino acids. Those amino acids get broken down into ammonia and urea. And the NH3 is then converted into urea because NH3 is quite harmful to the body. Urea is harmful, but not as harmful as NH3. Then we add water to the urea to turn it into urine. So that's how urine is produced. So our urine is full of urea. Urea, our body needs to get rid of. So blood containing high levels of urea is taken from the vein in the arm. The blood is mixed with thinners to stop clotting. The blood enters the dialysis machine, so it enters this machine where it is separated from dialysis fluid by a partially permeable membrane. So what we've got is blood on this side, blood, and on the other side we have the dialysis fluid. Now, what you'll find is you have the same amount of sugar on both sides. So that means the amount of sugar that goes this way, diffuses that way, the same amount of sugar diffuses back. You've obviously got more, urea on this side. So that diffuses across into the dialysis solution. So that's leaving the blood and going into the dialysis fluid. Now, the reason why the concentration of ions is kept the same is so that when the blood goes back through this second tube, it goes back with the same amount of sugar, same amount of ions, same amount of all the good stuff that was needed. But it comes back with no urea or less urea. In other words, with the body, the blood is removed, all the poisons, toxins are removed from the blood. So look, urea is removed from the bloodstream through diffusion, helping to restore the balance of water and ions. Now, dialysis fluid contains a glucose concentration similar to the normal levels in the blood, and an ion concentration similar to those that are normal in the blood plasma. And this explains what I just explained a second ago. Is urea produced by the body? It is. Protein, so the breakdown of dietary protein results in the excess of amino acids. Now, most of these amino acids are recombined to make a new protein. But sometimes people go on certain types of diets which are high in protein, which aren't an issue if you have a lifestyle that requires lots of protein, like someone who's doing lots of weights and increasing muscle, etc. But all the excess amino acids is essential for the body to eliminate this surplus amino acids. So excess amino acids need to be secreted. So what our body does, the amino acids undergo deamination. So the amino acids get converted into ammonia, NH3. Now ammonia is very toxic, so it's rapidly converted into urea, which can then be secreted from the body as part of urine because lots of water is added to it. So exactly what I was just saying before. Now that I've explained all of that, we can go back to this slide and look at advantages and disadvantages of dialysis and kidney transplant. Now, part of the issue with with dialysis is you have to keep a tube in the arm at all times. Because when you're doing dialysis, you go to the hospital 3-4 times a week for 4, 5, 6 hours. So you have this, wherever you take the tube through, there's a permanent kind of thing there to allow you to very quickly attach yourself to the dialysis machine. But the fact that you've got this hole in your body, or you've got this this area where you can add the tube in, it automatically, what does that mean? It means you've got chance of infection. So advantage of dialysis is, and usually a person has uses dialysis or has a kidney transplant if their kidney has failed. And we're not talking about just one kidney, we're talking about two. One kidney we can survive with. But if we've got kidney failure in two kidneys, then we've got a problem, or the person has a serious problem. So dialysis advantage, it keeps the person alive until a transplant is available. Downside, and also it removes waste, salt, and water excess, waste, salt, and water from the body. There's no chance of rejection. It's not a living thing that's entered your body, so there's not going to be any rejection. The issue is it's very time consuming. Having to go hospital, travel all the way there, stay there for 4, 5, 6 hours, and then come home again 3 or 4 times a week. That's a lot. And it means a person needs to take anti-clotting drugs. And there's a higher chance of infection. It's expensive for long-term use. And that's an issue because you can get sepsis, I think, almost like blood poisoning. And from an NHS perspective, it's expensive. For anyone that pays, anyone that's part of the NHS, which is everyone that lives in the UK, it's not expensive. Obviously, we don't have to pay for it, other than our taxes. Now, kidney transplant, there's no need for a regular dialysis. So it means you're not going into the hospital 3 or 4 times a week. It means a person can go back to leading a normal lifestyle. You don't take anti-clotting drugs all the time. However, when a kidney is transplanted into a person, it takes a while for the kidney to be accepted by the body because ultimately it's a new protein. It's a new thing that's just entered the body. It's the same as a white blood cell. No, sorry, it's the same as a bacterial cell or a viral cell, a virus entering the body. The body will very quickly see it as an outsider and try to destroy it. So there's a risk of rejection. It means the person has to take immunosuppressant drugs. So they've got to suppress the immune system. And there's a long waiting list. Donors are difficult to find. People who've got two kidneys are not necessarily ready to give their kidney up. Just in case themselves, they end up needing, they lose one of their kidneys or one of their kidneys loses function. So, topic 8 is animal coordination and control and homeostasis. So what are the examples of diffusion in animals? So oxygen diffusing from the alveoli into the blood, in the capillaries, that's one example. Carbon dioxide diffusing from the blood into the capillaries, into the alveoli is another example. Urea diffusing out of the blood plasma into the kidneys to be excreted is another example. So they're all examples of diffusion within the body. Now, carbon dioxide diffusing in and out of the leaf, oxygen diffusing in and out of the leaf, and water diffusing in and out of the leaf is also examples of diffusion. Water, also osmosis, which is a type of diffusion in and out of the leaf. So at the top of the leaf, as we know, transpiration occurs. As transpiration occurs, water enters in the roots through osmosis and it leaves through the stomata, which on the underside of the leaves, due to evaporation, so it diffuses out of the plant. And we've already covered the different conditions that affect transpiration. So more examples of osmosis in animal plants as well is absorption of water from the soil through the roots and movement of water in and out of cells within the body. As we know with red blood cells, if they fill up with too much water, they swell and they can burst. And if too much water leaves the red blood cells, then they can shrivel up and then they cannot transport as much oxygen. And active transport. So within the root hair cell, you've got lots and lots of nitrates and you've got lots of other minerals, and you have less of them within the soil. So naturally, what's going to happen is the minerals are going to start diffusing out, or their general movement would be outwards. But what the plant does is uses active transport to bring more of these ions back in to maintain a high concentration. Now, sometimes what can happen is if, for example, the soil becomes waterlogged and it becomes very, very dilute, that means the concentration of nitrates decreases even further because now they've dissolved into the water, and that makes that, and in that scenario, active transport needed even more. Now, whenever we say the word active transport, what should we automatically link it to? We should link it to mitochondria. And what is mitochondria? That's where respiration happens. And what does respiration provide? It provides energy. Another place where active transport needs to happen is sugar molecules are absorbed from the lower concentration in the gut into the blood. So this sugar can then be used for respiration. Okay, surface area to volume ratio. A single-celled organism, such as bacteria, have relatively large surface area to volume ratios. This facilitates the adequate movement of molecules in and out of the cell, ensuring the organism's needs are met. Now, if you've got something like bacteria, it's got a very big surface area. It's got all of that surface there to absorb all the nutrients it requires. And that's enough of a surface to fulfill that internal area. But when you get larger molecules, for example, not larger molecules, larger organisms, like multicellular organisms, for example, let's look at the example of a polar bear. If you look at the skin of a polar bear, that's the surface area. That is not enough of a surface area to fulfill the requirements of all the cells on the inside. So small animals, their surface area to volume ratio is larger. They have enough surface area to deal with the volume. But larger animals, like human beings, polar bears, etc., they have a smaller surface area. So their skin is not enough to absorb all the nutrients, all the water, all the oxygen that all the cells on the inside need. So there's more cells on the inside, less on the outside. Small organisms like single-celled organisms, there's enough surface area on the outside to deal with the inside. So it is adequate, it's enough. Whereas with animals and plants, they need exchange surfaces. That's why we've got lungs. That's why we have a small intestine, because this increases our surface area. So in a multicellular organism, the surface area to volume ratio is much, much smaller. That means our surface area, oh, sorry, that's not very good there. Let me try again. That's better. That means this surface here, the skin, is not enough to provide all the nutrients required for all the cells on the inside. So to facilitate the effective exchange of molecules, these organisms possess specialized surfaces designed for material exchange. What that means is we've got lungs. So that outer surface there is not enough for all the inside. So we need something on the inside then that increases our surface area. For example, the lungs. If you were to flatten out the lungs, it would cover around 2 tennis courts. So that therefore provides enough surface area. 2 tennis courts is enough surface area. That's enough cells to get all the oxygen we need to remove all the carbon dioxide waste that we've made and that we don't need. Our small intestines, again, they would cover a massive surface area if we were to get all the microvilli and flatten them out. And that is our method of taking our broken down food and putting it into the blood. So the blood can then transfer it to all the different cells in the body. So our lungs and our intestines increase the surface area. That allows for efficient respiration and digestion. Similarly, in plants, leaves have a large surface area. So leaves have a large surface area to enable optimal photosynthesis and gas exchange. So if you look at the leaf, they're quite thin, but the surface area is large and they're facing the sun. So that means they can absorb maximum of energy and also they're full of stomata, so lots and lots of gas exchange can occur. Okay, alveoli. So now we're looking at each one separately. So it's something animals have, particularly human beings have within our lungs to increase the surface area. So to increase the amount of oxygen that we can take in. So when explaining the adaptations of the exchange surface, you need to identify an adaptation, give a clear linked explanation to this, and to this adaptation to explain how it increases exchange. So you need to say what the adaptation is, and alongside what the adaptation is, you've got to give a clear linked explanation to that adaptation. So let's look at a 6-mark question. What are typical adaptations of the lungs for gas exchange? This is why 6-mark questions are so good. And it's important to memorize the answers. And this is why we've covered so many of them in the pre-exam masterclass. And this is why anyone that joined the pre-exam masterclass was given a booklet just full of 6-mark questions and 4-mark questions. Because when you memorize them, what you're actually memorizing is that whole topic or a big chunk of the topic. So look, the first thing we've identified, the lungs have got lots of alveoli. Why is that important? Because it increases the surface area for exchange. So it means the body takes in more oxygen and releases more carbon dioxide. Then it's got a good blood supply, which maintains a steep concentration gradient. So what that means is, um, let me just redraw this. So the pre-exam workshops will be on the weekend before each exam. So not this weekend, but the following weekend, there'll be 2 sessions, one for biology and one for chemistry. I know that Eid for many will be on Friday, and people will still be celebrating on Saturday and Sunday, but that Saturday and Sunday are important, it will be an important weekend for GCSE students. So I would advise that you enjoy the festivities on Friday and then you give up your Saturday and Sunday for your revision. So I think Saturday will be biology and the Sunday will be the chemistry workshop. But if you check the timetable, it will be on there. And unfortunately, I'm sure some of you have exams on Friday as well, on the day of Eid. But there's only a one-off, only one time that is going to happen. Right, so imagine the blood is flowing this way and it's coming with less oxygen. So what happens is, because the blood's flowing through, the oxygen enters here, then the blood continues moving. Then the next one comes without any oxygen, so the oxygen diffuses across. Once this red blood cell's full of oxygen, there's not going to be a concentration gradient, but then another red blood cell comes. Because it's flowing, new fresh red blood cells with a lack of oxygen keep flowing past. So there's always a concentration gradient. There's always a red blood cell that doesn't have any oxygen, doesn't have much oxygen. So it's a good blood supply and it maintains a steep concentration gradient. Also, well ventilated, which maintains a steep concentration gradient. Well ventilated means we're breathing in and out. So when we breathe in, we bring in more oxygen. When we breathe out, we remove the carbon dioxide. So that means we keep this full of oxygen and we keep emptying out the carbon dioxide from it. So carbon dioxide keeps moving from the blood into the alveoli, and the oxygen keeps moving from the alveoli into the blood. We've always got concentration gradient. Next thing, the walls of the alveoli are thin, and that provides a short diffusion pathway. So you've got four different adaptations, and you've got four explanations. It needs to be memorized. Okay, this is a triple only topic. The whole of this topic is not triple, there's just small sections that are triple. So 8.4b, factors affecting diffusion. So the first thing we've got here is temperature. So high temperatures will lead to a faster rate of diffusion. This is because the particles are moving faster. So if you've got a high temperature, it'll lead to a faster rate of diffusion because the particles are moving faster. An example of this is making tea. When you immerse a tea bag into hot water, it brews but much more quickly than in cold water. So if you've got your two cups, this is hot and this is cold. If you put your tea bag inside, this will change color much more quickly because the water's hot. It's giving the particles more energy, so it diffuses much more quickly. In cold water, it will diffuse but be much, much slower because there's less energy being given to the particles. Another one is, another factor that affects the rate of diffusion is concentration. So if you use the example of making tea again, if you add a tea bag that's got more tea leaves inside it, it will brew faster. So if you've got 2 cups of tea, same temperature, this one's got a tea bag that's got 1, 2, 3, 4 leaves inside it, but this tea bag here has only got one tea leaf inside it, or it's got 2 tea leaves inside it. Then this one's going to diffuse much faster. It's going to have a greater rate of diffusion because it's got more particles. Okay, for other factors affecting the rate of reaction is surface area. So the bigger the surface area, the greater the rate of diffusion. An example of this is the alveoli, which are found in the lungs. Their large surface area enhances the efficiency of gas exchange within the respiratory system. If you have a surface that's only this big, and you've got a surface that's this big, which one's going to cause diffusion to occur faster? It's going to be the big one. Why? Because in the little one, the gas can only diffuse across this small area. In this larger surface, you're going to have more space for more gas to diffuse across. Now, with the case of the small intestine, but in the what's called the alveoli, you also have a bloodstream here. So any oxygen that transfers across, what happens straight after that? The blood takes it away. So there's always a concentration gradient. 8.5b, this is still triple only, biology only. How can the rate of diffusion be calculated? Now, my advice would be we will be answering questions on this in the pre-exam workshop. My advice would be to look through some past papers and really have a quick go at practice some questions linked to this law. Because rate of diffusion is proportional to surface area times the concentration difference divided by the thickness of the membrane. Now, if a question like this comes up, it will just give you the formula and you just have to calculate the answer. Okay, 8.6, the structure of blood. This is a topic that's for everyone. So blood is a tissue. Now, when we say something's a tissue, what do we mean? We mean blood is a tissue. By tissue, we mean it is a group of cells working together. So what you've got here is the plasma, which is the liquid, in which that you've got your red blood cells, your platelets, and your white blood cells, all working together. And each one has a different function. You need to memorize these functions. This could come up as a 6 mark question or part of a 6 mark question. It could be a 3 mark, 4 mark, depending if it's higher or foundation. In a higher paper, it could come up as 6 mark question. However, it'll be part of something that requires a bit more thinking. So it's unlikely to be as simple as what are the four blood components and what are the functions. But this could just as easily on a higher or a foundation paper come up as a 2 mark question as well. So red blood cells transport oxygen around the body. White blood cells ingest pathogens and make antibodies. Platelets help with blood clotting. So if you have a cut, so there's someone's knee and there's the cut, the platelets start blocking up the cut to stop infection. Then you've got blood plasma. This transports sometimes waste products like carbon dioxide and urea. It transports digested food, hormones, and also thermal energy. That's why for those that are doing triple award, vasodilation is when the veins and arteries, the veins expand in size, particularly the ones by the surface of the close to the surface of the skin, so that heat energy can be lost. And on a cold day, then the veins become narrower, vasodilation, vasoconstriction, so that less heat energy is lost. Okay, you also need to know about each blood vessel. And what you need to remember is you need to know about their structure and also what their function is. So if you look at an artery transports oxygen-rich blood at high pressure from the heart. Now, the capillary is the site where molecules are transferred between cells and the bloodstream. So what happens is you have your artery, which is quite wide, especially the aorta. Then that splits into the arterioles, which are less narrow. After a few more splits, you get the capillaries, which are only one cell thick. Now, capillaries are only one cell thick, so that the nutrients that are being carried within the plasma, for example, glucose, can easily transfer across, and the oxygen that's been transported by the red blood cell can be easily diffused across as well. Then you've got the vein. The vein carries deoxygenated blood at low pressure back to the heart. Now, there's one scenario where all of this is different, and that is if we're talking about oxygen-rich blood. Now, oxygen-rich blood is inside an artery. Now, if this is the heart, arteries take blood away from the heart. So usually, not well, every circumstance like one is full of oxygen. Then whatever brings the blood to the heart, we call a vein. And that has a reduced amount of oxygen. Um, Actually, that's fine. I was going to give an example of the coronary artery and vein, but we can discuss that afterwards. Yeah, so the artery is the one that takes blood away from the heart. The vein brings the blood back to the heart. Then you've got the capillaries in the middle that are narrow and small enough to take oxygen to every single part of the body. Okay, now this is now the actual adaptation of each. So not just the function, but how it's adapted to fulfill its function, which is important for us to fully understand as well. So the artery, it carries blood back to the heart. No, sorry, it carries blood away from the heart. So it carries blood away from the heart and it is at high pressure. So it carries blood away from the heart and it's at high pressure, not low pressure. So if you have your notes in front of you, please correct this. So the artery is thick muscular walls to handle the high pressure. Because at this point, the blood that's entered into the left side of the heart is oxygenated. So it needs more space. So the artery has a small lumen, as in the narrow space inside, that's lumen just there. It's got thick muscular layer and it's got a thick elastic fiber layer as well. Now what happens is when the heart beats, the blood goes into the aorta, which is an artery. And as the blood goes through the narrow lumen, it expands outwards to deal with the extra amount of blood going through it. So this is why it needs the elastic fibers. Now the blood's going at quite a high pressure, so it needs the thick muscular walls to deal with that high pressure. But just like an elastic band, the artery expands and then it recoils and moves back inwards. And that expansion of the lumen, which happens because the elastic fibers, and then the recoil is what keeps the blood pushing. It keeps pushing the blood forward. Is why we have a pulse. That pulse is not the heartbeat, the pulse is the arteries expanding and recoiling. It's stretching and recoiling, stretching and recoiling. Okay, this is all 8.8. Okay, so the vein. This carries blood back to the heart. So this is correct, low pressure. There's a few things here. It's got thin walls because the pressure is low. So it doesn't matter if the walls are thin, the walls can deal with it. It's got a large lumen to help blood flow easily. And it's got valves to stop blood flowing backwards. So what that means is, usually with veins, for example, the vena cava, the biggest vein in the body, is taking blood back to the heart. So imagine a vein taking blood from the feet all the way up to the heart. And what's going to have an impact on the flow of the blood? Things like gravity. But every time the heart beats, that residual blood that's being released keeps the blood flowing. So now if you have these valves, and this is what they would look similar to, and you've got blood here in this section here, the heart beats and the blood moves, pushes the valves to a side, and it enters the next chamber. But as it enters the next chamber, it collides with the valve here and here. And when it collides with the valve, so there's your valve. As the blood's gone through, they've been pushed open. But now, what the blood is doing is closing them shut. So the blood pushes against each side and it pushes them against each other. So it closes them shut, stopping black flow. So let's look at that one more time. The blood flow pushes through the valve, and when it hits the top part of the valve, it closes them shut. So that means the blood can't go back to its previous place. Hey, the capillaries. So the capillaries, this allows exchange of substances. It allows the exchange of substances. For example, like oxygen and glucose between the blood and the body cells. So, like I said before, they have very thin walls. They're only one cell thick, the walls are. And there's only space for one blood cell to be passing through at a time. Now, it's got very thin walls, one cell thick, for a short diffusion pathway. So the oxygen can very quickly diffuse across and go to the cells that require it. And then carbon dioxide can very quickly diffuse into the plasma. It's very narrow, so only blood cells pass through one at a time. So you get one blood cell at a time. Okay, so why is the heart known as a double pump? So why is the heart known as a double pump? The heart is an organ that pumps blood around the body in a double circulatory system. So what that means is when the left side of the heart beats, so when the right side of the heart beats, let me change the color so you can see it more clearly. Whoops. So when the right side of the heart beats, it sends blood to the lungs. So when this side of the heart beats, it sends the blood to the lungs. When the left-hand side of the heart beats, you can see it's just slightly thicker there to deal with the pressure of the oxygenated blood. So the blood comes back from the lungs, the left side of the heart beats, and it sends the blood to the rest of the body. But these beats are occurring at the same time. So the blood arrives in the top chamber just here, the atria. Then both atria will contract at the same time. The blood will go through these valves. That's where the lub dub sound comes from the heart. When the valves open and close, lub dub. Then this part here fills with blood. Then they both contract at the same time. So the blood from here, the blood here will go through the lungs and get oxygenated. The blood here will go through the atria and to the rest of the body. So the right atrium is responsible for pumping deoxygenated blood to the lungs, and that's where gas exchange occurs. So it gets filled with oxygen. The left atrium, however, the left ventricle, um, yeah, so that's the left atrium at the top there. This bit here is the left ventricle. It pumps oxygenated blood around the rest of the body. So you have more muscular tissue with the left ventricle because it's oxygenated blood. Okay, these are four of the main arteries and veins. So vena cava is a vein, and it brings blood from the rest of the body. So vein which returns from the head, the neck, and the upper limb. So it brings blood from the rest of the body that is deoxygenated. It brings it into the right atrium. The right atrium then contracts and sends blood to the right ventricle. The right ventricle contracts and that sends blood through the pulmonary artery to the kidneys. Not to the kidneys, sorry, to the lungs. Once that blood reaches the lungs, it's oxygenated, it comes back to the heart. So what brings blood back to the heart? The pulmonary vein. The pulmonary vein then transports the oxygenated blood from the lungs back to the heart. When it goes, sorry, it doesn't go there. It would enter through here. No, apologies for I'm doing this the wrong way around. It was right the first time. So it enters through here. The pulmonary vein takes the blood to the left atrium. The heart contracts. It goes through the valves and it goes to the left atrium and then move into the left ventricle, which has its thick muscular wall to deal with the additional pressure because of the oxygen. The blood then, the left and the right atrium ventricle contract, and the blood is released into the aorta, which is an artery that carries blood from the heart to the rest of the body. So these four, it's important you know them inside out. So deoxygenated blood goes from the vena cava to the right atrium, the right ventricle, the pulmonary artery, and then to the lungs. Now, oxygenated blood comes from the lungs to the pulmonary vein, through the left atrium, into the left ventricle, then the heart beats again, and the heart is sent out to the rest of the body through the aorta. Okay, now what actually makes the heart contract? It's a group of cells in the right atrium called a pacemaker. So it's a group of cells in the right atrium, just here, called a pacemaker, and it controls the heart's resting heart rate. So it essentially releases electrical current. That electrical current moves right down the middle here, and then up through the sides. And as it does that, it causes contraction. It causes contraction of firstly the atrium and then the ventricles. So what is respiration? Respiration is the reaction that happens all the time in living cells to release energy. So respiration we've discussed quite a bit, but we're going to go into more detail now. So where does respiration happen? In the mitochondria. Respiration happens in the mitochondria, and that releases energy. And there's two types of respiration. There's aerobic, that uses oxygen, and that happens in the mitochondria. And there's anaerobic, that happens without oxygen and releases less energy. And that does not happen in the mitochondria, it happens within the cytoplasm. So what do we need energy for? We need it for movement, so we can run, walk, and move, keeping warm. Again, this topic, at this point here, this topic links back to topic 1 and enzymes. And it links to homeostasis, maintaining the same temperature, particularly in cold weather. And growth and repair. Energy is needed to help the cells grow, divide, and fix damaged tissues. So aerobic respiration is the complete opposite of photosynthesis. So if you know what photosynthesis is 6 carbon dioxides, 6 waters make glucose and oxygen. Respiration is the complete other way. It's now glucose plus oxygen makes carbon dioxide and water. Definitely memorize that. Now, when we exercise intensely, our muscles need more energy than the oxygen can supply. So we end up getting to a point where our cells that are contracting. So firstly, if we're running around, our muscles are full of muscle cells, and each muscle cell contracts and relaxes. Now, to contract and relax, it requires energy. So it's got lots and lots of mitochondria. When aerobic respiration occurs, lots of energy is released. However, if a person is really working out intensely, they get to a point where the oxygen gets used up, or there's not enough oxygen that can be supplied to the muscles. So they start using anaerobic respiration instead. Now, in anaerobic respiration, glucose is converted into lactic acid and some energy. But what the lactic acid does, it's an acid at the end of the day. It can cause some of the cells to start denaturing. That's why at the end of exercise, and those of you that are studying P will know this really well, at the end of exercise, at the end of exercising, the lactic acid will react with oxygen and it will be released as carbon dioxide. and water. That's why we have an oxygen debt. After we've exercised, we start taking deep breaths and start taking as much oxygen in as we can. That oxygen debt is there to get rid of the lactic acid, that oxygen that we owe the body. Okay, anaerobic respiration in yeast and plant cells both are living organisms. When yeast and plant cells respire anaerobically, in other words, without oxygen, they carry out anaerobic respiration, but instead of lactic acid, they actually produce alcohol and carbon dioxide. So our bodies during anaerobic respiration produce lactic acid, but in the anaerobic respiration in yeast and plant cells produces ethanol or alcohol and carbon dioxide instead. So anaerobic respiration in yeast cells and plant cells is glucose becomes ethanol, which is alcohol, plus carbon dioxide, plus some energy. Energy we don't need to mention because it's not a part, it's not made of particles. When we're writing a chemical equation, we look at the conservation of mass. So energy is not required. Now in yeast, this process is called fermentation. And it's the process that we use to make bread. And it's the process that we use to make alcohol. Well, not we, but some people will. Now, anaerobic respiration in yeast and plant cells, it needs, so this is a comparative, and this is, again, it's a very good, it's a very easy question to turn into a 6-marker. So you should know this properly. So aerobic respiration does it require oxygen? Aerobic, yes, it requires oxygen. Anaerobic, no, it doesn't. What about the products? Aerobic makes carbon dioxide and water. Anaerobic makes lactic acid in animals, but in plants and yeast, it makes ethanol and carbon dioxide. Now, the relative amount of energy that's transferred, the amount of energy that's transferred, aerobic transfers more energy because there's plenty of oxygen, the glucose is fully broken down. It's like saying complete combustion. Complete combustion releases more heat energy. Aerobic is, in a sense, is a complete combustion. It's a complete reaction, so it releases more energy. Anaerobic releases less energy because the glucose is only partially broken down. It's converted to lactic acid. Now, this is the core practical. So again, make sure you've memorized it. Make sure you can answer questions on it. Now, in the pre-exam workshop, not this weekend, but the weekend after, we'll be going through, as we have in every single session, we'll be going through lots and lots of required practical questions. So here, a respirometer. Also, what I would advise alongside what I've just advised, I've advised doing lots and lots of past paper questions linked to required practicals. I would also advise watching these practicals on the Edexcel Pearson website, on the YouTube channel, sorry, because they're going to go through it in a way that will benefit you for the exam. Now, a respirometer can be used to investigate the effect of changing the temperature on the rate of respiration. So here, you've got a mesh to rest an organism on. So you've got a small little insect on there. Here you've got soda lime to absorb carbon dioxide. So how can the rate of respiration be investigated? So a respirometer can be used to investigate the effect of changing the temperature on the rate of respiration. You place your live organism and non-living organisms in and compare the difference. So in one test tube, you might have dead organisms on the mesh, and this one you might have living organisms. Now, what happens is the soda lime absorbs carbon dioxide. So as these insects start doing respiration, they release carbon dioxide. that carbon dioxide is absorbed by that soda lime. And that causes, because it's being absorbed and taken in, it causes this little, in the respirometer, it causes this bubble of gas to move in this direction. And that shows respiration is occurring. Now, if it's a dead animal, there's no carbon dioxide to absorb, so the bubble of gas won't move. Okay, calculating heart rate. Now, with these kind of questions, like I've said before, we are going to practice with the math style questions, the numeracy questions when we do our pre-exam workshop. So we will have a practice of this question. But heart rate, as in how many times the heart beats per minute, sorry. Stroke volume is every time the heart beats, how much blood is being pumped out. And cardiac output is therefore how much blood is pumped out per certain amount of time. So if we've got heart rate, which is beats per minute, stroke volume is how much blood is given out per minute, then cardiac output is the overall amount of blood that's released from the heart per minute. So if someone had a stroke volume of, say, 250 milliliters, and they had a resting heart rate of 60, what we do here for cardiac output is 60, so 250 times 60. So that would be 15 liters. So an ecosystem is the interaction of a community of living organisms and the environment. So first thing we need to understand is what is a community? A community is made up of two or more different species that live in the same place. So it's two or more different species living in the same place. And then an ecosystem is the interaction of all those living things within a community, but it's the interaction with the environment. So in a sense, it's an interaction between living things and non-living things. So abiotic and biotic. Abiotic being non-living and biotic factors being living. So how is an ecosystem organized? You've got your producer, and producers are your plants and algae, etc., that have chloroplasts to help them photosynthesize. So they are the ones that are able to make their own food. They are eaten by primary consumers. Secondary consumers eat the primary consumers, and then you have tertiary consumers that eat the secondary consumer. Now, so the organization and feeding relationships in a community can be represented by food chains. Now, food chains, if you ever draw it within an exam, it needs to be straight lines. We've put some different types of lines just to make it look good, but it should be straight lines. And what those straight lines show is a transfer of energy. So it's the transferring of energy from the producer, which gets the energy from the sun, and it shows how it goes from one animal to the next to the next. So as we just said, what producers are the ones that photosynthesize. So they're taking carbon. When we say carbon, we mean CO2, and they're taking nitrogen. So that could be nitrates through the soil. It could just be nitrogen gas. They take, no, so not nitrogen gas, it's because it's compounds. And so it would be nitrates from the soil. They take it from the environment, and they use them along with oxygen, hydrogen, and other elements to make small organic molecules. Now, when we say small organic molecules, we mean sugars, fatty acids and glycerol, and amino acids. Now, amino acids are used to make proteins. So if we eat a plant and we take the amino acids from the plant, we then put them in a new order, as in we make a polypeptide. We combine lots and lots of these monomers, these amino acids, to make proteins. Fatty acids and glycerol are combined within our bodies to make lipids. And glucose, sugar, can be converted into starch. Of course, we need it for respiration. It makes the cell wall of plants, because cellulose is made from it. So sugar and sugars also are turned into amino acids. Because if you get your sugar molecule and you add a nitrogen group to it, which comes from the nitrates, you have now made an amino acid. So, as I've just said, these small molecules are subsequently utilized to create larger compounds, for example, carbohydrates, proteins, and fats. And what are the consumers? Consumers can only get their supply of carbon and nitrogen compounds by eating producers and digesting their biomass. So when we digest things like plants and animals, it breaks it down to small molecules. Those small molecules can then be absorbed into our blood and then they can be used for our biomass production. So they can be used to make more proteins, more carbohydrates, make more fats. So these are topics that I've already explained, that we've already covered. Okay, abiotic and biotic factors. So abiotic factors are non-living factors that affect the community. And biotic factors are living factors that can affect the community. So abiotic factors, for example, light intensity, temperature, all these things will affect a community, particularly when we talk about light temperature, when we talk about light intensity, we might assume it only affects plants. But if the light intensity is low, then we have a reduction in photosynthesis. And if we have a reduction in photosynthesis, it means we have less food. So it becomes a biotic issue. Less food, less plants grow, less food. And that then has a knock-on effect on all the consumers, all the primary consumers that eat the food, which has a knock-on effect on the secondary consumers and tertiary consumers. So light intensity, so abiotic and biotic go hand in hand. Temperature is another abiotic factor, moisture levels, soil and pH mineral content, wind intensity, carbon dioxide levels for plants, oxygen levels, pollutants. Now, biotic factors are living factors that affect a community, so availability of food. But it can be abiotic factors that cause the biotic factor as well. New predators arriving that can mess up the food chain, a new pathogen, one species outcompeting another, so one having a genetic variation that helps it survive. Now, light typically, so this is what I've already just explained, as light intensity rises or increases, the rate of photosynthesis increases. That means more photosynthesis, plants grow bigger, so that means there's more food for consumers. What I explained was the opposite way around. Now, temperature. Animals or plants are adapted to survive in particular temperatures. So if the temperature shifts or an organism is relocated to a habitat with a different climate, it may face challenges in survival. So at the moment, because of global warming, if we look at something like, for example, the polar bear is struggling to survive because the temperature is going higher and quicker during the summers. It means the ice starts to melt quicker than it did before. Now, when it didn't melt if we just go back 30, 40 years ago, the melt wasn't melting as soon. So what that meant was the polar bears were able to hunt for longer because polar bears can catch seals and they can hunt their prey on the ice much more easily than they can in the water. They are faster than the seals on the ice, but they're slower than them in the water. So that means now, across the summer, a lot of polar bears aren't getting enough food to survive. Soil pH and mineral content, so another abiotic factor. Some plants and aquatic organisms are adapted to survive in different pHs. So if there's a change in the pH, the organism will face challenges in its survival. So for example, now if we look at acid rain, when the acid rain falls, it can change the pH slightly of certain areas. So if you've got an area that's got a lot of acid rain, which is the case in some parts of the world, then that means the soil will become slightly acidic and some plants will survive and others won't. Carbon dioxide levels. So this is something that's needed for photosynthesis. The higher the concentration of carbon dioxide, the more photosynthesis will occur, leading to increased plant growth. And right now we're increasing the carbon dioxide levels because of different man-made reasons, which will come later in this topic. Wind intensity and direction. So lots of organisms prefer sheltered locations. And plants are more likely to settle and germinate in a sheltered location. So animals, generally speaking, live around those sheltered areas because they would expect more plant life, more food to be there. Moisture levels. Now, if water levels drop significantly, it can lead to the death of both plants and animals. Because, for example, a red blood cell, without enough water, it will shrivel up and die. It will shrivel up and it won't carry as much oxygen, sorry. And with plant cells, again, they become plasmalized, as in the water from the cell. They don't have enough water for the plant to survive, it needs to be turgid, it needs to be full of water. Because plants don't have skeletons. So our skeletons keep us upright, but in a plant, what keeps them upright is the turgid cells. So having water, being full of water is important. Now, when moisture is too high, the roots become surrounded by water, and roots are then unable to obtain oxygen. If they can't obtain oxygen and certain nitrates, or certain minerals from the soil, because it's waterlogged, that also reduces the respiration, can lead to that plant dying or ultimately perishing. So oxygen levels for aquatic animals. Water contains oxygen and is essential for the survival of organisms that inhabit aquatic environments. Without it, animals would suffocate and die. So contaminated water contains low levels of oxygen. So water that's contaminated has low levels of oxygen. And sludge worms are an indicator of low oxygen because sludge worms can survive in low oxygen environment, and that indicates pollution. Another example of this is eutrophication. So it does come afterwards in this PowerPoint, but I'll explain it now. What ends up happening is if you've got your pond and life is normal, but because of a local farm, and this could happen in a lake as well, because of a local farm, it's rained a lot and the fertilizers ended up in this small lake or small pond, and so you get an algal bloom, or the plants on the surface grow quite rapidly. Now, what that does is it blocks all the sunlight. So all of the plant life at the bottom that was providing oxygen, it starts to die. So when it starts to die, these plants die off. That reduces the oxygen being produced for the water. And also, what happens instead is the microorganisms start to feed. The microorganisms start feeding off the dead plants, and because now you've got lots of microorganisms there, they use up all the oxygen. That makes the water oxygen deficient, and things like fish die off. Now, if a question comes up in an exam linked to eutrophication, you don't need to mention eutrophication at all, just the process. So it's more of an understanding that this is one thing that can happen that reduces the oxygen levels within water. So then that means anything that's living in there, the larger animals that need to respire, they start to die off because they suffocate. They don't have enough oxygen. So more than anything, I'm just explaining this bit here. So what are examples of biotic factors? So new predators. One species are competing another. An example of that is the red and grey squirrel. Large availability of food and pathogens. Let's go through one at a time. So predators, this results in significant decrease in the prey population. So now if you've got one prey and you've got two or three predators trying to eat it, it starts to decrease that prey. But that has a knock-on effect because some of the predators that were eating that prey may have only had, that might have been one of its main food sources. So now if that food supply diminishes or decreases, that means that predator will start to decline in population as well. Now an example of a new predator being introduced was in Australia, where they introduced the red fox as a new predator, but that had devastating effects because it just meant the numbers increased massively and there was nothing to reduce the red fox population. There's quite a few examples in Australia where new things were added without thinking. So now, if the number of organisms that eat the food will increase, that means you can have a limit of food. So if the food becomes limited, sorry, so if there's a large availability of food, the number of organisms that eat the food will increase. But then if there's not enough food, the number of organisms will decrease. Pathogen, an example is Native Americans. In fact, there's lots of examples of South America and North America when the Europeans first went across there to colonize those countries or those countries within South America and North America, within those continents. They brought along with them European diseases. And because these the people who were the people who lived there, who were the natives, the indigenous people, the people whose land it was, they had never they had no immunity towards these diseases. So and they didn't have the genetics to save them in many cases. So a lot of people died from things like the flu or the common cold. A more recent example is COVID. And one species out competing another. The grey squirrel is a very good example of that. So a few hundred years ago, people started to bring grey squirrels from Canada. It was more than anything else. It was more of a showing off type thing, more of a flex to say, look how rich we are when we've got managed to get this special giant squirrel. Because the grey squirrel is massive. Well, it's a lot bigger than the red squirrel. So it was more to put into their home mini zoos, so to speak. But over time, these grey squirrels escaped or people stopped bothering. And now, grey squirrels have outcompeted red squirrels for food, for resources. And grey squirrels also carry a disease that kills red squirrels, but it doesn't kill them. So this is all. So that means the red squirrel population has decreased massively to the point that wherever they do have red squirrels now, they kill the grey squirrels so the red squirrels don't become extinct. Now, interdependence and competition. So competition is when different organisms within a community are seeking the same limited resource. And usually those same limited resources are food, mates, water, space to hunt, and space for shelter. So their habitats. Now, these are resources which are limited in supply. Resources are objects or substances that an organism needs to survive. Now, in animals, not animals, sorry, in plants, so I should say plants, they compete for light, space, water, and minerals. Now, interdependence is when species rely on each other for survival. So usually within an ecosystem or within a community, you've got multiple animals and each one has a reliance upon the next. This is a really good example that plants rely on the bees for pollination and the bees rely on the plants for food, sometimes for shelter as well. So certain animals that have this kind of this interdependence with plants, they provide the plant, they help the plant do with their seed dispersal and their pollination, but in return, the plant provides food, shelter. Now, this example here of the bee, the flower depends on the bee for pollination in order to generate its offspring, but bees rely on the nectar and the pollen as a source of nourishment. But this would have knock-on effects. So if we had a lack of bees, that would mean we would reduce the pollination, we'd have less plant offspring, less plants would mean less food for other insects as well. also less of a, it would reduce the habitat for certain insects. So it all has a knock-on effect. So what is a stable community? So generally speaking, a stable community is when the population, so we've just said a community is made up of lots of different species. So it's got, and each species has a population. So um, within one community, you've got multiple populations. You've got different species with their own population. If that community's numbers roughly stay the same, then you would call it a stable community. So a stable community is one where all the species and environmental factors are in balance. This means the population size remains fairly constant. So if a change occurs within a community, it can have significant consequences for all inhabitants. Because everything is interdependent and the one thing has a knock-on effect with something else, it means that as soon as you as soon as one thing is impacted, it has a knock-on effect on the remainder of the community as well. So mutualism is a relationship in which two species benefit from their interaction. So a bee and a flower is an example of mutualism. This is where both species take benefit from each other. Whereas parasitism, so another example of mutualism is the clownfish and the anemones. Clownfish possess immunity to the sting of anemones, allowing them to seek shelter, keep shelter from creatures to evade predators. So the clownfish is able to survive within the anemone because it has immunity to its sting. So for the clownfish, it provides shelter and protection. In return, the clownfish eats the parasites that might come and settle on the anemone, keeping them, and it also provides them with nutrients through their feces. Now, a parasite is a relationship where, so mutualism is where both species take benefit, but parasitism is where only one takes benefit. So one lifts off the other, it benefits by the expense of the host as well. So the parasite extracts what it requires from the host, yet the host gains no advantage in return, and in some cases it can lead to illness. So parasites are generally adapted to extract as much as possible from their host while ensuring that the host remains alive. Okay, so this is a core practical. What I would advise is if you go to the Edexcel, Pearson's YouTube channel, they have examples of all the different practicals, which are core practicals. So my advice is I will go through this now, but please go through it there as well. So what you do here is, let's say you've got a plot of land that is 10 meters by 10 meters. So in other words, it's 100 meters squared. And you want to know how many daffodils there are within all 100 meters squared, roughly. So what you do is you throw a quadrat like this, and this quadrat is 1 meter square, 1 meter by 1 meter. So it is 1 meter squared. Now we can't put 100 of these down to fill this whole space. So what you do instead is you throw the quadrat with your eyes closed into random spaces. You throw 10, or you throw it over your shoulder. Or what you can do is on a piece of paper, you split this into 10 by 10 squares, and then you label everything. So you might say this is A, B, C, D, E, F, 1, 2, 3, 4, 5, 6, 7. And then you'd use a random number generator to create the coordinates. So it might be B7. So you know that's over here. Put your quadrat there. You might have F3. So you know you're going to put your quadrat there. That kind of thing. So, and by doing that, it avoids bias and ensures results collected are valid. Now you throw a 1 meter squared quadrat randomly about 10 times. Find the average of the dandelions in 1 meter squared. So if you've thrown it 10 times, time number one, you might have counted 20 dandelions. Time number two, you might have counted three. Time number three, you might have counted seven. The fourth time might have been 22. You add them all up and you average the amount in 1 meter squared. So if 1 meter squared had 12 dandelions or daffodils, that means on average in your 100 meters squared, you just multiply it by 100. You would have had 1,200 dandelions or daffodils. And you can do that in different areas. So you might do it initially in the shade, then you might do it in the sun, and then you can do a comparison. So if you look at the picture here, you can see it's got a sunny area, it's got a shady area. So you throw 10 quadrats in in the shady area, 10 quadrats in the sunny area, and then you can compare your average, or you can multiply it up by the total area of the whole thing. And then you've got a rough idea of how many dandelions or daffodils or whatever plant you're looking at is across the whole area. Now, the other thing you can do is use a transect. So you place a 100-meter transect line starting the edge of the field. So you might start from here and have this piece of rope or string run for 100 meters. For example, if you started it here, it would run through the woodland as well. And then every 10 meters, you put down your quadrat and you count. If we're using the dandelion example, you would again look at the dandelions. And using the transect line helps investigate how light affects the distribution. Because as you're going through this line, you count the number of dandelions, sorry, is going from bright sunny areas to dull kind of shady areas. So those shady areas, the shady areas won't have as many dandelions, so you can do a comparison. Okay, so field work techniques. So this is something I've already explained, where you just multiply it up to find the whole area. So please spend time and memorize this practical. It's a very common practical that comes up in paper 2. But when we do our pre-exam workshop, we will be covering all of this. Okay, so what are the different trophic levels? So this is 9.7b, so this is purely triple only. The whole booklet is not triple. So once we've gone through 9.7 and 9.8, 9.9 is back to being combined and triple again. So anyone that's doing combined, you only have to be patient for 5-10 minutes. So organisms such as plants and algae generate their own food. These organisms are known as producers. So it's something that we already understand. And what happens is the producers take in sunlight energy. Now, usually the amount of energy they take from the sun is about 1%. 1% of the sun's energy. So now, but what we're going to look at here is we're going to say plants have got 100% of the energy. Now, the primary consumer eats the corn, eats the plant or whatever it is. In this case, it's bananas. The fruit flies eat it. Now, they waste, not waste, but they use up 90% of the energy in excretion and in movement. So we're talking about things like respiration, excretion, movement. Again, when the toucans eat the fruit flies, that is our secondary consumer, they use up about 90% energy, again, in excretion, in respiration, in movement. And then when the jaguar eats the toucan, it uses up about 90% again in the defecating, so excretion into the feces, in respiration, etc. Now, because what actually happens is, on top of that, is when we eat. So, for example, we eat something, a big chunk of it is fiber. So it means we don't even access that energy from the first from the start. So if you're eating an apple, a lot of it is fiber. The skin is fiber. All the cell walls in the cells that are inside it is all fiber. So it goes through one end, it comes out the other. We don't take any of that energy. Now, biomass pyramids can be constructed to illustrate the relative quantity of biomass present at each level of a food chain. So when we say biomass, it means how heavy, how much it weighs, or what the mass of it is. So we always have more producers than we have consumers. Now, producers, primarily plants and algae, transform, as I said before, 1% of the incoming light into usable energy through the process of photosynthesis. So that 1% of light that's absorbed is converted into the chemical bonds in the glucose. Now, trophic level 1 is at the bottom of the pyramid. Now, how is biomass lost between each trophic level? Only about 10% of the biomass from one trophic level is passed on to the level above. So only about 10% is passed on from one step to the next. 90% is lost. Now, why is 90% lost? Because not everything that's eaten that's ingested is absorbed. Some is egested or removed as feces or excretion. Some absorbed material is expelled as waste. Carbon dioxide is released back out. Water is lost during respiration, as well as water and urea, which is found in urine. So if we eat, for example, if we eat an apple, as the example was before, a lot of that water will be excreted from our body. Okay, so you can work out the efficiency of energy transfers by biomass input minus the biomass output divided by biomass input. So you calculate the difference between the biomass between the two trophic levels. You can also do this for energy to calculate the efficiency also. Okay, back to triple and combined topics. So what are the problems of fish farming? So first things first, what fish farming is, it's a really big net. Baby fish are released inside it. Those fish start growing. Now, if that's the net, the fish will stay within that net, and the net will go quite low down. But at a certain point, it won't be all the way to the ground. So you have the normal ocean floor underneath as well. So fish farming involves large nets or tanks within lakes or in the sea. Fish farming generates a significant amount of waste, chemicals, pathogens, and parasites that are released into the surrounding water. So certain fish, like salmon, require a higher protein diet and can be fed wild fish, which may lead to a decrease in the local population of the wild fish. So there's a few issues with this. That when the fish defecate or excrete, all the excretion goes down to the bottom here. So all the feces are at the bottom here. On top of that, the fish food that isn't eaten ends up in the water underneath. And again, you've got decomposed that will come and try and eat it. So what ends up happening is this part of this ocean or sea or lake ends up with a slightly different ecosystem that can have a knock-on effect on the other fish. And then that can have a knock-on effect on the wild population as well. But one thing this does do, it does allow, it stops people from fishing and it keeps the wild population higher. Okay, then you've got non-indigenous species are introduced. So human beings have been doing this for a long time. They've been introducing themselves in other people's countries. And causing the indigenous population to decrease. But this happens with animals as well. So a non-indigenous species is one that does not naturally occur in a specific area and may be introduced by humans for various reasons, such as pest control or hunting purposes. So a lot of this kind of stuff's happened in Australia when they released a lot of toads, but those toads didn't reduce the problem, and the toads had no predators, so the toads increased in number massively. So the example we know of already is the grey squirrel. The grey squirrel outcompeted the local red squirrel, and now there's not many red squirrels left. A new species has the potential to outcompete or kill native or naturally occurring species. That leads to reduced biodiversity and can also lead to extinction. So this I've already explained, eutrophication. Water can become contaminated from multiple sources, including households, factories, and agricultural operations. So household waste can taint the water, while chemicals utilized by farmers might wash into nearby bodies of water and cause eutrophication. So you can see the eutrophication there underneath the bridge. That is an algal bloom. So what is biodiversity? Something that's really, really important and really, truly is something that should be studied in more detail outside of just GCSE because it matters so much. It's something that's so important. And the better we understand it, I mean, it will lead to a better understanding in the next generation and the generation after. So it's the planet's ours. It's important that we look after it. We shouldn't see ourselves as one individual. We should just see it as it's our responsibility to look after it. The same way it's our responsibility to look after our homes, to keep our own bedrooms clean, etc., to look after ourselves. We should be treating the earth in exactly the same way. So biodiversity refers to the variety of different species of organisms on Earth or within an ecosystem. So it refers to the variety of different species. Now, the more species you have within an ecosystem, so an ecosystem is how living organisms interact with the non-living parts of the environment. An organism is just a living thing that has organized structure. So in an ecosystem, it's about how living organisms interact with each other. Now, the more species, the more different types of animals, insects, plants you have within an ecosystem. So the more biodiverse it is, the more stable it will be. Because it means each of these organisms have multiple sources of food. They've got it's not a food chain, it's a food web. Because it's got so many different species involved, it means if there was to be an issue with one of them, it won't really have too much of a knock-on effect. So the future of humanity relies on maintaining a diverse and healthy ecosystem. Both plants and animals provide us with essential resources. It's our duty to safeguard species for the well-being of future generations. Many human activities are decreasing biodiversity, and only recently have efforts been made to stop this decline. So, what is deforestation and why does it occur? Deforestation occurs when trees are removed from a large area. We do this to create space for cattle. Now it says rice fields, but more accurately it would be farms. It's not fair to just target those countries that have rice fields. Lots of countries have cut down trees for cattle and farms. It also allows for the cultivation of crops intended for biofuels. So some countries are cutting down trees so they can grow certain types of plants that can be turned into fuels for cars and for running other things. Which is a problem because really, if you've got an 8 billion people population, 8 billion plus population, land should be used for food. So the advantages of deforestation are it allows to grow crops in farms, rice being an example, to use the land for grazing animals like cows, and to grow crops for biofuels. But on a disadvantage, now you've got fewer trees to take in carbon dioxide. That means more carbon dioxide in the air, so that can lead to global warming, massive loss of habitats, and there's less biodiversity. Now, what do some humans do to try and reduce the negative effects of what we have? So biodiversity refers to the different types of plants and animals living in the world or within a specific area. So one thing some farmers have started to do is restore field margins. So if, for example, someone's got a farm and in each section they were growing something separate, traditionally there were hedgerows separating each part of the farm. Now, what a lot of farmers did, they cut down these hedgerows because in one respect it makes sense. They want to make more money. So if they cut down the hedgerows, you've got all the extra space to grow. But what some farmers are starting to do now is they reintroducing these hedgerows because those hedgerows were habitats for lots of different types of animals. Then we have conservation breeding initiatives for endangered species, particularly in zoos. We're minimizing deforestation around the world. There's quotas for each country, so countries don't cut down as many trees. Also, there's a lot of countries now planting a lot more trees. For example, Saudi Arabia. Saudi Arabia has a plan that I think by 2050, 2060, they want to have planted 10 billion trees within the desert. I think they've already planted several hundred million. So, and that took them at least 10 years. So they've got a long way to go. And what do some humans do to try and reduce the negative effect that we have? There's now more protection and more restoration of rare habitats. Recycling resources instead of sending them to landfill is another thing. Because then we're not using, by reusing them or recycling resources, for example, with metals, with the example of metals, we're not having to quarry and break into as many mountains, ruining as many landscapes and habitats in the process. Okay, 9.11b is food security. Then 9.12 will go back to a topic for everyone. So when we talk about food securities, so this is triple only, 9.11b. It says this 9.11 is by itself, 9.12 will be combined again. So when we say food security, do we have enough food to feed the population? So some of the things that are being done, we have an increasing birth rate. So we do have a higher population. So we do need to secure our food. Agricultural input is becoming more expensive and there's new pests and pathogens. So changes in the environment can affect food production, potentially resulting in challenges such as famine in certain countries if rainfall is insufficient. Now, the way they're dealing with these food securities is, for example, if you look at these chickens, the chickens are put in environments where they don't use up as much energy. What that means is they're put in cages. So each one is put in a separate cage. And by doing that, the chicken doesn't move around as much. It doesn't use up as much energy. So more energy goes on growth. And now that does provide more food, but it is cruel to the chicken. And it's something that people protest on a regular basis. Although at the moment, people are protesting other things that are more important. This was a common protest, and people who are involved with the who are involved with these kind of charities linked to treating animals well, chickens are something that comes up quite often. It's they're not given enough space, so they end up having quite a horrible life until they are killed and fed to people. Also, from a food security perspective, we've already covered fish. There's a lot of overfishing happening, so there's a lot of species of fish which are heading towards extinction. But they are trying to increase the fish farms so as to not affect wild species. Okay, 9.12 material cycles. Why is it important that materials are cycled? Now, the carbon cycle transfers carbon from living organisms back to the atmosphere in the form of carbon dioxide, which plants utilize during photosynthesis. The carbon and water cycle both cycle materials so that they can be used as the building blocks for future organisms. Now, The water cycle provides fresh water for plants and animals on land before draining into the seas. Water is continuously evaporated and precipitated. So let's start with the carbon cycle. The easiest way to understand the carbon cycle is by looking at it from a perspective of what puts carbon dioxide into the air, into the atmosphere, and what takes carbon dioxide out of the atmosphere. So respiration, which is this process, glucose plus oxygen makes carbon dioxide, and water releases carbon dioxide into the air. Combustion is the burning of fossil fuels, so that releases carbon dioxide into the air because usually we burn hydrocarbons. When we burn a hydrocarbon, we make carbon dioxide and water. Decay is when something dies, for example, a plant or an animal, then decomposers will come and feed off that animal and they'll multiply in number because they've got so much food. And when they multiply in number, firstly, they're moving carbon compounds from the dead animal into their own cells. Then they're using those carbon compounds to multiply. And then each one of those decomposers also respires and releases carbon dioxide. Consumption just refers to the fact that when we eat something, we transfer carbon particles, carbon atoms from the thing that we just ate to ourselves. Now, all these different processes take put carbon dioxide in the air, except photosynthesis, which takes carbon dioxide from the air. So it'll take all these carbon dioxides from the air, and what it'll do, it'll react the carbon dioxide with water, take energy from the sun to release glucose and oxygen. So where on earth can carbon be found? So carbon stores are a place where carbon and its compounds may remain for a time. So carbon is locked inside fossil fuels, animals and plants until the animal plant dies. Sedimentary rocks, for example, calcium carbonate. Now, and what happens to a carbonate when it reacts with an acid? We know this from chemistry. What happens is it releases carbon dioxide. This carbon dioxide locked in the atmosphere, within the soil, and within the oceans. So photosynthesis takes carbon dioxide out of the air. Respiration puts it back into the air. Decay, because we had an increase in the number of microorganisms, they start respiring and releasing carbon dioxide back into the air. Combustion is the burning of fossil fuels, so that releases carbon dioxide into the air. And consumption is the process of organisms eating each other. That means carbon moves from one organism into another. Water cycle. So here we've got our collected water. So here we've got our collected water. The water initially evaporates. As that water evaporates and diffuses into, well, it rises upwards. As it gets cooler and cooler, it starts to condense until it forms clouds. And when the clouds have enough water inside them, precipitation occurs. Um, precipitation is the release of um, either rain. Precipitation is rain, snow, sleet, any of these processes. And finally, another example we have here is transpiration, which is water that's in the ground enters the plant. And how does it enter the plant? It enters through osmosis. It makes its way up the xylem, and then it diffuses out of the leaves. And that diffused water will also start to make up the clouds. So how do we obtain potable water? First things first, what do we mean by potable water? We mean water that's clean enough to drink. So we have 3 steps. Number 1, we pass the water through a filter bed to remove solids. So we filter. The second step, that filtered water is put in a big container, and this is sedimentation. So all these small particles that were too big to get stopped by the filter bed, they make their way to the bottom. And then finally, we sterilize using ozone, UV, or chlorine to kill the microbes. So usually in the UK we use chlorination. Chlorine is added and the chlorine kills the bacteria or any of the microorganisms. So filter, sedimentation, chlorination. Usually that's the foundation question, but it can be part of a more difficult higher question as well. Now, the other way we can get potable water is from the sea. We can do desalination, which can be done by distillation. So we can get our salty water and evaporate off the water or boil off the water and then condense that water, leaving behind the salt. Or it can be done through reverse osmosis. But both of these methods use lots of energy. Now, the way reverse osmosis works is like this. You've got your membrane. Now, on this side of the membrane, you've got lots of water. You've got lots of water on this side, and you've also got water on this side. But the side that has the seawater also has salt. And because it has salt, that means there's less water. In reality, what happens is this, the water attaches to the salt, so it becomes unavailable. And so what we've got here is a high concentration here and a low water concentration here. In other words, we're dilute here, concentrated here. So which way is the water going to go? Dilute to concentrated. But we don't want that to happen because we're going to end up with more salty water. So what actually happens is this, in reverse osmosis, almost like a plunger type thing, it pushes this way and it forces the water through. It forces the water through, leaving the salt behind. So now all on one side we've got pure water with some possible minerals, and the other side, what we're left with is the salt. Because the salt cannot get through these gaps, but the water can. Okay, so groundwater, we've got 3 steps. We filter, sedimentation, and chlorination. With salty water, we can use reverse osmosis or distillation. Now, decomposers, including bacteria and fungi, they play a vital role in breaking down and decomposing dead organisms. Decomposers carry out respiration, and that releases carbon dioxide into the atmosphere. Mineral ions are also released into soil. So when decomposers feed off something, they respire, so they release carbon dioxide into the atmosphere. However, they release minerals into the soil. Okay, nitrogen cycle. So what happens during the nitrogen cycle? Nitrogen fixing bacteria, so that's shown here. Let's use the highlighter, show it clearly. Just there. Nitrogen fixing bacteria transform atmospheric nitrogen, so that's N2. And remember, between the 2 nitrogens, what do we have? We have a triple bond. So it transforms atmospheric nitrogen into nitrates found in the soil. So that's one way of taking nitrogen out of the air and into the soil. Now decomposers, they break down dead organisms as well as urea. Urea is found in the urine and feces, and they break it down to ammonia. Then the ammonia, so what we're saying here is nitrogen fixing bacteria take nitrogen from the air and put it into the ground. Then plants can use that the nitrates that they've formed to make proteins and grow. What decomposers do is they convert dead organisms, urea and feces, into ammonia, but then nitrifying bacteria turns the ammonia into nitrates. So between these three different types of bacteria or microorganisms, nitrogen fixing, decomposers, and nitrifying bacteria, nitrogen is being converted into nitrates. So you need to memorize this because a very common question where well, I've seen a question just a few years ago which asked about the three different bacteria that are involved in taking nitrogen out of the air. And these are really good in higher paper. These are this would be make a really good 6-mark question. So what happens during the nitrogen cycle? Under certain conditions, the nitrifying bacteria convert nitrates in the soil into bacteria. So this is the bad one. This one takes nitrates in the soil, and the plant wants to use those nitrates to make proteins, but the nitrifying bacteria converts it back into nitrogen. Okay, so how can crop rotation also be add nitrates to soil? So crops such as peas, beans, and clover have nitrogen-fixing bacteria in their roots. So in the actual roots, they have these little nodules full of bacteria. Now they're nitrogen-fixing bacteria, and what do nitrogen-fixing bacteria do? They take nitrogen from the air and convert it into nitrates. So this is a very good mutualistic relationship. When growing, these plants, nitrates are added to the soil. So peas and beans and clover, if they're grown for just 1 year, so for example, 1 year you grow something that uses a lot of nitrates, the following year you grow peas, beans, and clover. In that year, the peas, beans, and clover replenish the soil with nitrates. And we've also got fertilizers. So natural fertilizers, including manure and compost. Compost can be applied to fields to provide a valuable source of nitrates. So you have fertilizers that have nitrates, phosphates, potassium, but nitrates are the ones that help with growth. So an increased concentration of nitrates in the soil will enhance crop yield because it has more nitrates, it's going to do more proteins will be made, and it's going to grow faster or grow better. Artificial fertilizers are also available for purchase and can be added to the soil. These fertilizers typically contain ammonium nitrates. So ammonium nitrates look like this. So it's got twice as many nitrogens. Okay, 9.16b, 9.17b, and 9.18. These are all triple only topics. So I'm going to go through them now. And after that, we'll be taking a break. So anyone that's doing combined, we're going to restart our physics at 4:15. So if anyone wants to go for an earlier break, we're going to restart our physics at 4:15. So 9.16b refers to indicator species. So what's an indicator species? We gave an example before. There's certain types of insects that grow better in poor oxygen water. So that would be an indicator of pollution. So indicator species can be used to determine the level of pollution. Indicator species serve as a means to assess overall pollution levels in both water and air. So some lichen do not grow well in the presence of sulfur dioxide. So if you see less lichen on trees, it suggests that sulfur dioxide levels are elevated. So lichen do not grow as well in the presence of sulfur dioxide. So if you see an area, if you see trees where there's not much lichen, that suggests there's lots of sulfur dioxide in that area. Rose black spot also does not grow well in the presence of sulfur dioxide. Now, fresh water shrimp and stonefly indicate that the water is clean. But bloodworm and sludgeworm, as we went through it before, indicate water is polluted. These are just other insects that you don't necessarily need to know the names, but it just gives you an example. The mayfly represents low levels of pollution, whereas something like a worm or a hog louse or bloodworms and sludge worms. Well, when it says worm here, that's referring to bloodworm. These indicate polluted water or water that has less oxygen inside it. Now, what are the ideal conditions for decay? Decay works better at high temperature, well, not a high temperature, but a moderate temperature when it's moist and the availability of oxygen. Now, when farmers, for example, put their composting, well, anything that's going to compost into the compost box, what that actually does is because microorganisms are breaking it down and releasing energy, it warms it up, and that warmth helps the decay happen faster. So temperature is important. Now, one thing you need to keep in mind here is that the decay is done by decomposers, which are microorganisms. So they're protein based. They also have enzymes which they release. So both the microorganism and the enzyme can denature. So at cold temperatures, decomposing organisms are less active, and so the rate of decomposition remains low. This explains why we keep food in a fridge or a freezer. So at low temperature, you've got low rate of reaction, as in the particles aren't moving fast. So think back to chemistry. The particles aren't moving as fast. They're not colliding as often, so you have less of a reaction. So the enzyme and the substrates don't come together. So that's why we keep food in the fridge or the freezer. It stops it from decaying. Now, as the temperature increases, decomposers become more active, and so the rate of decay increases. But at very high temperatures, decomposers will be killed, and so decomposition will stop. So at a normal temperature, up to a certain temperature, an optimal temperature, activity, the rate of decomposition increases. But beyond that temperature, at very high temperatures, the decomposers will start to denature, as in will die because of the high temperature, but also the enzymes will start to denature as well. Now, water. What a lot of these decomposers do is they secrete enzymes onto decaying matter, and then they absorb the dissolved molecules. Now, that would not happen without water. So that having water is essential. And the availability of oxygen. Decomposers can't survive when there's little or no oxygen. They need the oxygen for respiration. And that means when things are being decayed, as we increase the rate of decay, we also increase the rate of respiration. So we increase the rate at which carbon dioxide is being released. Now, the rate of decay is affected by temperature, water, availability of oxygen. So this means ideal conditions for decay are warm, moist, and plenty of oxygen, which is what that box would provide. So gardeners and farmers strive to create ideal conditions for the rapid decomposition of organic waste materials. So the compost generated serves as a natural fertilizer for cultivating garden plants or crops. So putting compost in a bin or a pile helps keep everything moist and warm. So having it all within that one box, whenever they're respiring, they're releasing heat energy, so it helps keep it warm. They're releasing some water as well, which helps keeps it warm, keep moist as well. And that enhances oxygen. Now, to enhance oxygen availability, some farmers or whoever's doing the composting will make a few holes, or using a garden fork or spade, they can just keep rotating the mud every few days. And that just increases the amount of oxygen within that compost bin. Now, 9.19b. How can the rate of decay be determined? So don't forget, when we say something has a rate, it's either the mass of it divided by time, so that's going to be grams per second, or it could be the volume of it divided by time, so that would be centimeters cubed per second. So to find the rate of decay, you would measure the starting mass of the material. You would then leave it to decay for a fixed time period, such as a month. Then you would reweigh the mass. Then you find the change in mass, and divide it by the time taken, and that gives you the rate of decay.